Tobago. Today we're diving northeast of Trinidad and Tobago in their waters, but on top of what is the largest accretionary prism on the planet, and that is the Barbados accretionary prism. So in this part of the world, you're not going to see a lot of seamounts or things like that. All of the topography is related to squishing of sediments as they are scraped off of one plate and scraped onto another plate, and the folding and faulting that goes on as a result of continued accretion of sediment from one plate to another.
This is not coming from me.
Huh. Alien? Could be. I'm hoping. There's a lot of evidence here of past life. These carbonate crusts and all of the tube worms and maybe clams or mussels. I can't tell what those shells are. Yeah. Depression. Yep. Yeah.
Stuff.
No, you found two red dots in 10 minutes. Yep, so we're coming up on another seep that was seen on the multi-beam this morning. We did a drive-by.
A little beefier here.
Yeah, this one looks a little beefier. So the darker mussels are the ones who are currently enjoying the feast on methane, and the lighter colored ones are past generations. Yeah, this is definitely the biggest. Okay. Okay, yeah, let's spend some time looking around. Yeah, cool. Yeah, so that white dusting is methane hydrate. Yeah, we can do a little bubble hunting. Cool. Yeah. Yeah. They're like cool. Look like like a statue that's been encrusted with mussels. Where is he? Oh, yeah. Nice. Okay, we got a big crab hanging out in the back, eating something, probably a mussel. Or a Sammy, yeah. Oh, look at all the shrimps hanging out under that, yeah, under the overhang there. There's like hundreds of them. Yeah, he's definitely eating a mussel. Num num num. That's a different crab than we've seen. Yeah, yeah.
Like this.
Yeah. This crab.
He looks like he's like gonna
Yeah, let's hang out with the crab for a minute.
bust open that muscle.
See if I can
It looks like he's already eating something, isn't
Real
he?
jelly.
Yeah.
Yeah.
Isn't this cool though? Look at all the shrimps in the
Yeah, the shrimp hanging out. It's like a stadium full of shrimp.
So it's the first two reptiles that we've seen on top of the ridge.
Can we get any closer to this
Oh, yeah. I'm
this
I'm
guy?
settling down.
Oh, cool. Okay. Yeah.
I just gotta get a little softer.
I mean, how does he pick which one he wants to eat, given when there's like thousands and thousands?
He's probably pretty sick of muscle.
Mussels again. Ugh.
Shrimp, Jesus Christ.
What's he gonna say?
Stretching.
Is stretching or it's yoga he's
So
doing?
this is another big new seep, huh?
Yeah.
Yeah.
See the
It's the
side of
biggest
it?
one we found
Yep.
on this site.
Definitely.
I'm just gonna throw it out there, Diva. You have an opportunity to name this Schmidt Ocean Institute.
Oh God. Oh my God. You have
Your
an
dad jokes
endless
is terrible.
supply of dad jokes over here. Zach just churns them out effortlessly. Look at what is he doing? Oh my God. What?
It's just doing the one side.
I don't know what's going on. Yeah.
You know, just hear him.
Why is he covered in sediment?
And look at all the sediment on him.
Didn't do it. I remember we were talking about something's kicking the sediment up. So I wonder if there's a big bubble
Yeah.
forming somewhere in this.
Yeah.
net, 'cause
That would make sense. Because this is the really fine grained, like, muds and clays. There's little shrimp everywhere. I don't see any of those eel pouts, though. Hmm.
okay.
Oh, another little crab? Squat lobster? Wow. Oh, there's a little squat lobster. Just tons of shrimp. I believe it's a squat lobster. Yeah, can we get the lasers on just so we can see how big these or
They'll be
small?
out of the picture until I zoom out.
Oh yeah, don't worry about it. Wow. Yep. Cool. Oh yeah, that's big. Is Diva still here? She left. We're wondering if there's something kicking up all this sediment, like maybe a bubbly seep. Maybe we can look for it.
We were
Around here?
Yeah.
flying around earlier and there's just like clouds of it going.
Oh, yeah?
Yeah.
Yeah, It's
Alright.
run out.
Keep going.
Yeah. I mean, it looks like it keeps going that way, right? Or
It does here too. No.
all right. Very cool. Not so little. Yeah. He's big. Yeah, you can see all the tube worms all welded together with this calcium carbonate. Whoa. Okay, this is really big.
Well, it looks like it's kind of transitioning. into
Yeah.
dead stuff now.
Might be, yeah. Maybe turn, see if we missed anything the other way.
Sure. Cool little crack there.
That is a cool crack. Wow. Yeah, that's. I'm guessing there's like bubbles coming out of all those white spots, right? Because that's where the methane's popping up.
Crabby?
So many. Oh, another crab. And another crab. I mean, why wouldn't they be here?
You know what's weird though? It kind of seems like the mussels and the tube worms don't really like each other.
Yeah.
They must have zoning.
It's like one or the other. Yeah, like zoning restrictions, like housing.
Tube worms are getting too long here.
Right. Those tube worms are crazy. Yeah. Nobody wants to live next to the tube worms.
Tube worms.
Wow.
There's shrimp all up in there too, man.
Oh yeah. Huh. Wow. Yeah, I'm not seeing a lot besides the shrimp and the mussels, but nope.
All right.
All right.
On to the next bubble.
On to the next bubble. Tube worms are the HOA presidents. Yeah, there's a hierarchy here. Yeah, we can head over the. Maybe that next dot is connected. No, probably not. That would be too easy. Oh, another big crab.
These are shrimps. You want a marker here or anything?
Yeah, should we put a marker here? I'm out. Oh,
Yeah.
what did
Can
I?
you go to the meeting with the links to decide tomorrow? I'll cover you for.
Oh, okay. What's the meeting?
They're like deciding what to dive tomorrow.
Oh, okay.
So
I was
they're
like,
in
did
the
I say
conference.
something wrong?
No.
Was it about the tube worms? Oh my God. The tube worms got together and got me fired, you guys. Okay. I'm out.
Yeah, they're in the conference.
Okay.
Tomorrow. and
Yep.
We're needed.
Okay, perfect.
Bye guys.
Howdy. How are you?
I'm good. I'm good. You all stuck with me again. This is Giuliano. Rachel got Sumon to help defining where we're gonna dive tomorrow.
So we just finished that little red bubble. Now we're gonna go to the one that we.
Cool. She did not got fired, by the way. Just clarifying.
Yeah.
Yet. Yeah. Yeah. Although those were her last sentence in here, she's not right. She will be back. So did we end up dropping a waypoint in that previous one?
We did.
Okay, cool. Thanks, Zach.
We called it big mussels.
Big mussels sounds about right. Yep. And lots of crabs.
One crab guarding this little mussel trap still.
Yeah, that was
Get off my lawn.
yeah, look at that. I think we've seen at least three or four different species of crabs, at least visually speaking, during this cruise. Or at least that I can remember. These white ones, the red long ones, some very spiky spider crab likes, and these big ones that we saw in the previous batch. We're continuing our search for bubbles. Yeah.
not too far from the next red dot. Probably pretty short move.
Over there, is it?
Yeah.
Bridge, Irving.
RV bridge,
Have an updated position.
update position.
Thank you.
Hmm? This looks promising. Ish. What is that? Yeah.
You just look to my left real quick. Pretty much on it. Anything that way.
Seems a bit everything like disperse. Can we just maybe do like a
Definitely not anything right on this.
no?
Right on top of that thing. No, there's not anything to the west. That's where we came from. Alright, where would you like to go?
So if we can do just like a quick loop around here just to see if we don't find anything, and if not, we can head down to point number 3
Sure.
just to make sure there's doesn't seem to be. But they all seem quite dispersed, this little areas. No. All right. So then I think we can head down to waypoint number 3.
3, if we're going with those red bubbles kind of under there too. Straight.
Yeah, kind of like those. Yeah, if we can like direct ourselves to those over there.
Bridge, Irving.
Want to fly over big mussels on the way?
Yes, please.
Are we
If we can, yeah, that would be a good one.
Can
Yeah.
stop the
Right
vessel.
on the way.
Nothing.
Stop.
Last point.
In a minute, they're gonna give you a new updated
Going
position.
back
It's gonna
to-
change the direction to west.
Last
Okay?
waypoint.
Okay, just let me know when you want me to start the bridge.
Always interested in fish, I am. This guy.
The old Subaru then.
Look at this
This is a weird one.
guy. Yeah. It's a good size. Maybe 60 centimeters. No. You'll like fish, but not more than that.
Bugs my life. Just having an
Fantastic.
adventure.
Yeah, that's all right. We can continue.
Okay.
Thanks, Doc. Do you know if we've collected all the small worms that we wanted? All the big ones?
The long ones will be after the
So we want more of the long ones. So we can try to do that operation.
Sure, I will warn you though, that firebox is pretty full of deep.
The BB14.
Not too sure how many more are.
Okay, it says here X3.
Give it a hack.
Yeah, so I wonder if we can try to find small ones of the long ones, because then we can put them in BB2B.
Almost as much on the left.
Yeah.
They go in here, mostly dead.
Oh, can we zoom into these bubbles? Look at that.
Bubbles.
Look at that.
Boobless. You want to stop or just
Yeah, let's stop and
stop?
thanks, Zach. Let's stop and look
Good amount
around.
of bubbles there, huh?
Yeah, it's a good amount. Look at that.
Bridge is moving. Go ahead. Stop in there.
That's it, not moving.
Not moving yet. Okay. Just can't stop finding cool stuff here, man.
Yeah,
Good problem to have, I guess.
it is a good problem. Exactly, good problem to have. Too much to see.
Do they want that sponge?
I don't know. That is a cool sponge. Have we seen that type of sponge? You wanna? Do you have your phone with you? Shirley's in the. Shirley. Megan. See that sponge. Look at that. Happy with the bubbles, Rob?
I like the bubbles.
All right. And we might actually have already collected this sponge before. What else do we got there? We got some mussels. Oh. Yeah, same way we already have. Great. So we already have that sponge. So that's great.
Okay. What now?
All right. I think we can continue.
You want a waypoint?
Sure, yeah, that's a good point. Let's put a waypoint
What do you want to call it?
and let's call it uh, I think small moss. How big is these waypoints? Can we call it a pink sponge?
Yeah, you got the pink sponge.
Yeah, that will mean I will remember that.
All right, carry it on.
What's the name again?
Pink sponge.
Pink sponge.
I see that pink. I'll just make a note to myself. Thank you.
Ready? Ready for the vessel?
Yeah, I'm gonna go quick by this big mussel spot on the way to it. I'm gonna fly by. So I'm gonna go a little to the north instead of and I'll go south again.
Oh, fishy on the move. Oh yeah, beautiful
No.
job. No.
Cute, huh? That's a cute.
Well, that is a jelly. Another fish in the top left of the screen.
Where are you doing?
Go ahead, RV.
What undertaken position?
Update waypoint.
Dun dun dun. So did we find bubbles in this one?
No,
No bubbles.
no, just about everything but bubbles.
I'm back.
You're back? All right.
Yes. Can you go
Rachel
see
is back.
Teresa's thing,
Yes.
please? Okay. I said I would, and I was like, I have to go back.
Uh,
Another one.
Yeah, we found bubbles and I named them.
You named them.
Pink. I mean, just like at the
You
waypoint.
named them?
The waypoint. Just so
Okay.
we know the waypoint.
Okay.
And so now we're going back to number 3.
Pink sponge bubbles. I
Yeah.
like the name.
We're
Okay.
We're
so,
going back to point number 2, waypoint 3.
okay. Perfect.
And we just passed.
So we're gonna do a little check mark over it.
Yeah, we did like the 3 points. there,
Nice.
and then we're
Okay.
heading back.
Sweet. Thank you.
They bailed you out.
What's that?
They bailed you out.
Yeah, we just had to make a decision about next dive. Cool. So look at you finding all the seeps, man. You're like 4, 1, 2, 3, 4. All
That
right.
one we just, that one red bubble we just went through there, that was a bust.
Oh, all right. You can't win them all, man.
No, there's another one.
This is looking good.
You did
Okay.
find bubbles.
I saw it was in the conference room.
It's funny though, where the bubbles were, there wasn't that much stuff.
No, just bubbles.
And there's a little bit.
I wonder if this is a younger seep. Hmm.
Waypoint on this one, or don't you?
Do I want to what?
Waypoint on this one?
Yeah, we can put a waypoint here. Do I have to name it?
You can name
I
it. Or you
mean,
can let me name it.
I'll let you name it. That works. I'm not feeling very creative at the moment. If something, if it comes to me, I'll tell you, but.
Yep, ready.
Muscle beach.
All right, so we're heading to the one Under 3.
Yes. Two boublés under 3.
2 bubles. Oh, there's a little lobster. Crab?
You see a lobster?
Nope, crab. Maybe.
Could be a crab-shaped lobster.
Could be. This little guy.
You can zoom on with it.
No, he's okay. We'll leave him alone. Oh, there's another one over there. Hiding.
All right, carrying on.
All right.
Some more muscly stuff over here.
Cool. Oh, it looks promising.
See the sparkles?
Yep. Yeah, it's cool. All that aragonite shows up as like reflective. Whoa. Yeah, that's another big one, hey?
It's not even on a red bubble, eh?
It's not. What's going on?
Oh, jeez.
Oh, geez. Are you becoming Canadian? All the
Sorry.
A's?
Sorry.
Sorry. Oh my God, I think he's making fun of me. Okay, so this one is looking good. Was not on the map. Not predicted by our bubble detection devices. Ooh, look at the crabby down here. Working his way up the pile.
Yeah, look at that guy out there.
Oh God, this guy is really big.
The guy eating. Some of these shells are huge.
Yeah. And they're all nice dusting of methane hydrate. That is a big guy. Okay, can we zoom in on? Yep, he's eating too.
What do they eat?
I thought they ate mussels. I mean, why would they be in a pile of mussels? They weren't eating them. Did these crabs eat mussels?
Some of the animals.
Yeah. He's like cleaning his.
Can you zoom in and see what's.
Yeah, let's zoom in on him and see what he's doing. Looks like he's got. Yeah, he's eating something. Oh.
Gastropod.
He's eating a gastro.
Is
Something in a shell?
it? Get in there.
It's a gastropod.
Oh. Wow. Okay.
Give it a park so I can
Yeah,
see
can we see? Yeah, I'd like to watch this for a second.
Park.
Sorry?
Whoa.
I'm gonna keep the vessel for a moment here.
What's that?
I'm gonna keep the vessel for
Yeah, that's fine. We're not going too much farther, right?
Oh.
It's ready to fight.
He might be ready to fight. He put his food down.
Such a beautiful little sea.
Yeah.
think
I
they're
love
spot
this.
on.
Like
You
Above
think
the
they're
map,
what?
100%.
You think what?
The Rob said the best ones we visited have been these.
At
three.
the along
And so
yeah.
on the rim
Yep.
might be, and so potentially to go over here.
Yep.
might hit these ones on the
Yep.
rim.
That's our trajectory.
Yeah.
He
Yeah,
had a
such nice-
gastropod.
I was
and-
wondering what he was eating.
he was holding a- Yeah, I don't know where. I don't know where he put it. He was just holding it.
He's also really sedimented.
Yeah, they've all got that same dust that's like
Yeah, exactly.
collecting in between the patches.
Yeah, these are lovely little communities.
It's interesting. This guy's doing one side too.
The other side though.
Yeah, the other
Yeah.
side. Is he
Huh.
like right-handed?
I don't know. I'm not sure. Where did he put his food? He just was holding a gastropod. I think he knows he's being watched.
Whatever he was doing.
Hmm.
He's like salty too.
Yeah.
There's some really cute calling awards. I got you. Yes, ma'am.
I love the colors.
I know. No,
Pretty
it's
good desktop
beautiful.
wallpaper.
It would be.
Camellia.
No eel pouts though. I'm kind of sad about that.
There's a those. I'll double check James paper as well.
Scale one?
Yeah, really cute little scale worms around.
Really
Like,
hairy.
look at it. Oh my god.
Oh wow.
So they got
And pause the vessel for now, please.
Oh, what is that little thing on the?
two.
What's this?
That's a polychaete, a worm.
Got it.
A scale
Scale worm. Okay. Oh. Looks like a
In an
brush.
ideal world, you would get well, Ladino 1.5.
These kind of
Like,
seeds
try
are
to
also
get
fun. The Where's World of the
Oh, I know. Every time. Cool.
You gonna keep looking or keep trucking?
Oh, we'll keep trucking to these
Oh,
two.
and the husband
Oh.
of the,
Oh, an eopout. Good. I mean, I don't know why I'm so excited. These are my new favorite. Oh God.
This looks just like Falkor.
They do. Oh my God. Yes. Oh, the luck dragon. Yep. No, he's shy. Oh. Wow.
So beautiful.
Okay. Pew, pew, pew. He's out.
Okay,
All right.
now we go.
We okay to go to the next one? Or do you want to chill out here for a bit?
No, I think let's just like get a good look at some things on our way out.
Yep. Yep.
All right. So we're taking off.
Yeah, we'll take a look around. Maybe get a
Just
Fly
around,
over.
fly around.
yeah.
But we're back to Childressy here, right? This is in Boomerang. So it is interesting why different communities, different mussels at different sites.
What are the smaller ones called?
These are Gigantitas childressi, whereas the other ones are Baphimodiolus boomerang.
Boomerang. Okay.
This is a nice batch. It's probably less than half the size of that
Looks
last
like a
one.
boomerang.
Yeah. Yep.
The last one.
Way
Maybe.
bigger.
Oh yeah, that's true, maybe. Oh yeah, no, there's both here. But it is interesting about like why some are dominant versus others. Another big crabby up here.
Mm-hmm.
Some shrimp under there too again.
Yeah. Under
Yeah, those
this.
shrimpies.
They're all hanging out under this thing.
Yeah. Oh, look at how they're all jammed into those cracks on the
Not big
rock.
on personal space. Right?
No. Yep. Another one. Wow.
Any other requests to look at stuff?
I think we're good to go to the next spot.
Red bubble 26F.
Red bubble 26F, exactly.
Read early.
Smiley Bridge.
Can resume the vessel move.
Reanimated.
Yep.
Oh, who's up next? Oh, hi. So we are exploring for the rest of this dive, but we're also following some information obtained from our flyovers with the ship, where we were mapping, and some of the plumes that were identified from those data. So we're heading right now to a couple more points that are suspected seep sites. And yes, we are the first people to see any of these sites, and also the first maps that have been produced by a ship for any of these sites. So it's really cool to wake up in the morning and look at the data and see all the structures that were previously just sort of flat spaces. All right, so I'm going to turn it over to Genia. I stole the pen last time. I'm leaving it here. Yeah, we're just going to stay on the high spots there.
Hi everyone. My name is Eugenia Thomas. I'm a MPhil student at UE studying deep sea woodfalls and deep sea debris. And I will be narrating for you for the next couple hours. And so as my colleague mentioned, this dive is primarily investigating seeps. So we have been mapping bubbles for quite some time, and this dive is basically to locate those bubbles, locate potential seeps area. And these areas have never been explored and also has never been mapped. So we have come across some already. And so we're just looking for any more we can possibly find. So I've showed you this many of times. So methane seeps are formed from when organic matter or animals that are decomposed within the sediment, and over time that decomposition builds up to form methane within the sediments, and eventually the pressure and the heat causes cracks on the seafloor surface, and when that happens you have methane escaping in the form of bubbles. And so that's primarily the synopsis of methane seeps environment. And you have many different organisms that are associated with these environments.
Very good.
So on this frame here you can see many dead mussels, in previous footage you also saw tube worms, shrimps that are living within the tube worms. You also saw mussels. And so ultimately, you can use some of these organisms as a way to detect if there are any methane seeps nearby. Oh, here's a skate.
The next one we see.
Zooming out.
One thing that is always constant in each of our dives are the sargassum on the seafloor, which is not surprising given its high population within the region. See a couple starfish here. I'm also seeing small mounds of bacterial mats, which is another indication of methane seeps or chemosynthetic environments. Oh, what that? Oh, octopus. And it looks like a Dumbo octopus.
Flippy flapper.
Yeah, they have stuff.
Mhm.
You want it?
Oh my god. I so enjoy looking at these octopus. They are like the most adorable species. Oh my god, that is so adorable. Oh, oh. And it glides so seamlessly through the water column. Oh my god.
Thing looked like
It is so cute.
It's coming directly towards me.
Yeah, it's like it's going directly towards you. He's definitely not shy, you guys. Like he's really putting on a show for us. Wow. And you're going to see every angle of the octopus. Reminds me of like this floating teddy bear.
This is like a flying gun drop. You could park, but I think he's probably gonna jump up.
Mm. And like he's literally coming towards us.
He's chasing me. That's aggressive. Hey, Shirley.
RV bridge,
You can pause the vessel, you know?
coming to a stop.
That is literally like the cutest thing ever.
You gotta try to keep that. Nice there.
Yeah.
Now I can see you. Pretty cool. Did you get some good video of that, Jules?
Yes.
Can I change the lights one time?
Yeah.
See if it's better or not?
Sure.
Still like jiggly too, right?
So guys, again, we are watching a flapjack octopus.
Is that what that is? It's a flapjack.
Yeah, the ears are small. Yeah, it's small.
Well, I'm happy when y'all are happy.
Yeah, we can go.
And on?
Yeah.
Later, bud.
Alright, so we are going to continue exploring, but it was nice seeing this cute and adorable octopus called a flapjack. So again, we're going to. Oh, this is. So we are literally bubble
RV bridge?
hunting.
Resume the bridge.
Seeing some dead and some living mussels as well.
Whatever it does.
Yeah. It's in place.
So if we see some of the tube worms that
Mhm.
seem to be alive and easy to get, we'd want some of those.
Alright.
Were you here when you were collecting the tube worms? So you're up to the expert now.
I can tell you that our buy box pretty full of those things. We're good to go.
Oh, okay. So all of them, the two of them were here. Yeah.
Mm. So you want more tube worms?
So but the thing is that they are too big and
Oh.
it's kind of full.
It's kind of a weird map.
Yeah.
So
So some of the coloration on the sediment is actually bacterial mat as a result of the high amount of methane within the water column.
little bubble.
Small jellyfish.
Vote whenever you're happy there.
Yeah. So we're following the red spots that are on this edge.
Okay.
They seem to be the active ones. So that's why
Oh,
we're going.
so the yellow is where we
Yeah,
reach.
we're going to the
Okay.
next to the next one.
Okay.
Okay. Shells over here.
Oh, this one's big.
So,
Yeah, this might be that pretty big.
so I think we're coming across a huge mussel bed.
Yeah, this is a lot right here. Alright, you just want to
It's
go fly
really-
around, get the extent of it.
Mm-hmm.
Your bubbles?
Oh yes, and I think we're seeing some
A lot of
bubbles.
bubbles. Lot of bubbles. Not big, big bubbles. Yeah, where there's not
Oh, we're
mussels.
seeing a lot of bubbles.
Oh, neat. Alright.
And again, these bubbles represent methane that would have escaped the sediment through cracks or fish fixtures within on the seafloor. And so even though we have mapped particular points where we would expect bubbles along our route, for example here, we spot some bubbles, some huge mussel beds. So we're just doing a quick exploring. Mm. And so I believe we're seeing two types of mussels here. The Batamilios mussel as well as the Gigotondus mussel. And so these are two common mussels that are associated on methane seeps. You've seen a lot of bacterial mat forming on the mussels as well. Sometimes they are heaps, so again, this has never been seen by anyone, so this is a really huge moment.
It also kind of proves the methodology y'all use, huh?
Mhm.
Find
Yeah.
things, you know you can do it again.
Yeah.
This is a whopper.
And again, huge thank you for our scientists who stayed up hours just mapping these areas. This is a lot.
This is as cool as They're all like
It is
3D here too. They're not just flat.
Yeah.
They're like on
rocks, so yeah. This is very densely populated.
the sound canal, see how far this goes.
Seen some shrimps as well.
I guess I could fly around the border every time.
Yeah, that's what I'm saying.
Yeah.
You think this one is bigger than the last one, right? I mean,
Yeah,
like the
this is the
other-
biggest
yeah.
one at this site
Yeah.
for sure, I think.
Yeah, so exactly.
The one before, the site before this, I know, that was a pretty big.
Oh, that was a big one.
This guy's pretty old. Alright, so fly around the edge?
Tripod. Yes.
Yeah. Think that's a tripod fish below there.
Another one landing like we saw before, right?
Yeah. Yeah. Definitely a tripod fish. Oh. And there's a starfish within the midst. And interestingly, though, I'm only seeing the yellow starfish, not the other types.
It's fine, the children see animal. So just remember when it comes in, like make sure it's fine, make sure
So again, two different type of mussels, but we're more seeing the Batomedius mussel. Also seeing some huge crabs as well.
that's great. Crabs, oh my god. All in the road.
Yeah, there's a lot of really
Mhm.
interesting stuff here.
Yeah. And earlier on we saw a crab feeding on a gastropod. And so you see these mussels, they're not just on the sedimented seafloor, that they're also on the rock formations.
You see all the bubbles earlier, Beagle?
So two here and then two here and then two. No, I'm going down. Oh, yeah. Yeah.
So again, lots of bacterial mats.
Something to write on my map. On that a little bit real quick, just tell me.
Some more bubbles.
Why wouldn't it use to be? I would
Tons of bubbles.
But again, like if we're putting another bed, then we can be more time in the ones that we want for.
All right, so you see on the map
Other
there,
dogs tomorrow.
that donut I just did?
Ah, yes.
That's a mussel pouch.
Do you want to know that I would all?
So you can kind of guess.
Then we will regard.
So,
as the back to the
prefer-
picture there.
each one's a meter.
Over
Each of those squares.
Some
is
of
a
them.
meter.
Okay.
this, I don't know what.
Yeah,
Yeah.
stuck in here.
Oh, Mr. Smooth is here, okay?
Zoom on the fish.
What's that? Oh.
Boy, did you show up at the right time, buddy. Let me take this turnout, so I don't leave you in a bad way. All right, buddy, enjoy. It's cool here.
the size of the circle that we drew on the map?
Yes, right.
Take a picture.
Yeah, you can take one. Oh, what was sorry?
No, yeah. So you were saying the circle that we draw was sorry, this was the size of the square there?
So I flew around the outskirts of it. If you wanted to estimate
Yes,
the
please.
size, so each of those is a square meter.
Okay.
And so that one.
Okay. So we can take a picture of that.
Yeah. See this little one there? Not that. That's when I turned around. But this one is shaped like.
Okay. This one.
Yeah. I mean, I could just send it to you.
This one.
Okay?
Thank you.
So this was the largest mussel bed we've seen for the entire dive, and so it's really, really cool.
So you will collect some mussels here. 5 and 5 we find the other ones. But first with these ones that are more common.
The bottom video.
This is the other one. I mean, we
The
have
guy.
both, but most of them are the other one. The
The
one that
guy.
is not looking for.
One to the-
You can see that.
Yeah.
She bless you.
So you want to collect 5 of them?
Yes.
Anywhere special?
Anyway, we could collect it here.
Anywhere special you want to
No,
go?
just 5.
Doesn't matter.
Yeah, that's enough. 5 that are alive. Yeah, and yesterday we saw the bigger ones, but most of them were dead. They seem to be alive. You see what's close, and they were dead. So just have to make sure that they are alive.
We can tell that if we see it's called the lip. The initial.
Yes.
Yeah. I forget the correct name. I forget the correct name. The lip was
Cycles?
yeah.
Is that right?
These look dead.
These are things, right? Roof.
Probably if you go further in the middle, these look dead as well.
These on the rock, dead as well.
Those look alive. So
You don't want those.
yeah, we want the living ones. So these look alive.
So you want those?
Yeah,
Or
the
one on
living
the
ones.
ground?
No, the ones on the ground are dead. Yeah.
Some are alive, some are dead.
Yeah. So these for sure are living. Yeah.
You get those.
Yeah. So those in the heap are for sure living.
So good. Anything.
Yeah. What did you use the last time? This book? Yeah. So especially where the shrimps are, you know for sure those are
So this one.
alive.
Got it for that.
So I got at least 5.
shrimpy that has green eggs in his head? Oh look how they're eating this mussel. Whoa! Yeah, so he's a different species. So if at any point we could sample him. Yeah, another one here. Then for example, then
What tree? That's tree.
and the plan
Tree
is
and one's very
we're
small.
working on the extension now.
Yeah, just one is fine.
I'm just feeling out whether we'd have enough pulp seeders. But the plan would be to ping pong,
Okay. Which means get the box.
which means like yeah, like bam, 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 until we like
Sounds
run
fun.
out of time. I think sampling is less of a priority unless we see anything.
Yeah. Yeah, we're getting these.
Once we get
I can
these-
get these ones now.
Yeah, we get
Okay,
the back
shrimps.
boomerang for her. We can try to get some troopers or get a shrimp,
Big box.
but it's more if we like see like
I haven't seen
reverse
the
stuff.
boomerangs in these.
Yeah,
So you
and
could
it's
put it
bad,
maybe?
sugary. But also, I know it's hard, but we'll try to find some scale.
Right, box tree.
The ones. Yeah, they said that this one is basically free.
So we can
Because they were like, yeah, that's what it's like the things are this is food. And I think we collecting the food ones, we should be less this one. Try this and that too.
I mean, we'll probably see it. Yeah. Because didn't we already get two?
Hey, what?
Yeah, but they were like this large.
Tree.
So
Pigeon tree.
because they were like,
Pigeon tree.
Yeah, so I think if we could try to get some more.
Challenge.
Go in.
yeah.
Put it right here on the side.
Yeah, pigeon. That
You say you want to
big web.
collect the shrimp as well? Or the
Yeah, it's,
Yeah.
I've seen those. It's the one that has the green.
green that- yeah.
Yeah. So at least a couple of those.
How much kimo?
Huh?
How much kimo?
Much chemo?
Yeah.
6.
6? Okay.
You want more just in case?
No, five was the least, so that's good. Thanks. And then we're also trying to collect the shrimp that have green.
We're gonna slurp that?
Yeah. So if we can find it back.
Was that shrimp?
Yeah, but it has like green coloration on top of its head. Let's see it again.
Shrimps? you said?
Yeah, the one with the
Yeah. Probably.
that green coloration. Is that eggs? Was that eggs? The green coloration.
Maybe I'll say that it might be a different species. But
you want to put it in this? I think I'm gonna have to move. I have to put the porch.
Oh, I think I see one of them.
Okay, keep them on side if you have.
I think I'm gonna have to move that side. Or you could find
Can you
it.
get two Niskins at the site?
At this side?
Trying to see Niskins from places like this that don't have sponges, but like that kind of did it for anyway.
Okay, other one.
So are you seeing the strip?
No, I'm not seeing it again. So it have a green coloration on top of its head.
This one here?
Is it this one?
It's green on the head.
Oh yeah,
It's
but
green
this
on
one
the belly.
green on the belly. You wanna collect that one? So that one have a green coloration. on
No.
it. Just one inside head
Green
alone?
here on the head.
Uh, oh, look at here. This is it?
Yes.
Yeah, yes, that one.
So what's the green?
Eva says it might be a different species, but I also.
Let's see if you see any on this side. Because I have to move.
Well, that's a chance to get a better view.
Uh-huh.
It's different inside.
Just try to come up first and watch it. Or the strap. The little trap. Something on it.
I think is this the other
That's
one?
another one.
This is the other one here?
Yeah.
Yeah?
Is this one here?
Yeah, that's
Yeah.
another one.
Hey,
Try
this
it
here
out.
better. So we haven't suction anything yet. So I guess we'll play into
One.
one. Yeah. That other one by himself here.
Yeah.
That one might be easier.
See where it is? Right.
Yeah, yes, without this one.
Release this, yeah.
Yes.
No,
No.
look, it's still there.
Oh, oh.
It's going over.
um.
One here. He's coming back out.
Oh, yeah.
Zoom into where we were, just coming back out of the hole.
Oh no, look at one come back on top here.
One on top.
Yeah.
Top. Yeah.
Ah, nice. Yeah. I
No.
think
Move a little faster than you go that. And that's it. Go for it. Go for it. Up.
oh.
Jump. Come down on it. Kind of couple it. Squeeze in.
Okay, no, that's going in the hole. Ah, yeah. There you go. Yes. Yeah, I think we got it.
Oh,
Yeah, one more.
you got one?
Yeah, we got one. You want
Let's
to
try
do
one
that?
more.
Hmm? That is one. One.
Okay. So you got that. That one has sex, but now they have.
The muscles wasn't.
Yeah, yeah. But if I put right here, also the
So
number.
what? The arm down.
So it was 375.
That's for the muscles, right?
Yeah.
Just remember, fitting section example.
376.
correct.
Which one?
I have one right here. But that one might be too hard.
This one here to your right.
Alright.
This one.
Oh. Okay, oh. See?
There's also two on that direction as well.
Suction up. Yeah.
Oh, nice. That's it?
There's another one just at the right of the. Yeah, it's not on the sea. No, it's behind this load.
Which one?
Yeah, it's just behind the suction sampler.
You wanna move
Take
out
it
this
away.
suction sampler?
Yeah. You see it? Yeah, right there. Yeah, right.
Got it?
Yeah, we're done. Thank you.
Hmm? Right, so he wants to do two bottles in this side.
Huh?
So the best thing for this one bottle one more.
Sorry?
Rob said he wanted to do two Niskin bottles at this site. So should I
Yes.
just
Just now, yeah.
Okay.
Finish with
Yeah,
it?
you can
Take
do
it
that.
one
Yeah,
time?
finish with the suction.
No, this area. Finish with this
No.
area.
No, we need
You
to
will-
take two Niskin bottles.
So for returns, every time use number one just to clean the tube. So every time we collect a sample, it shouldn't be
No,
here
no, no.
because it has a the size of the mesh is bigger.
Mm-hmm.
So
So
you
put
should
it
start on 2, then 3, 4, you know.
Mm-hmm.
So we use this one just for
Wait, for Niskin?
research.
Yeah. Yeah.
Yes.
Yeah,
2
nice.
Niskins.
Before y'all take off from here, can I just look at one thing for like 20 seconds?
Yeah.
Are y'all about to
Yeah,
take off?
Niskin. Yeah. You want to go for it?
No. I just
Wait,
wanna
you
move
have
the
time?
camera real quick if you don't.
Definitely fire 1 or 2.
Two?
We need 2.
Yeah, then.
Fire 1 and we need to fire another one.
So two bottles, right?
Two?
Yeah, two.
Good afternoon and good evening, everyone. My name is Zahir, and I'll be taking you through the next hour of our and the last hour of our dive today. Currently, we're just going to deploy 2 Niskin bottles to collect some water for some eDNA analysis. Hi, everybody. Happy to be here with you. And that was our second Niskin bottle being fired. So we're on an interesting site here. Quite a bit of diversity, a lot of tube worms, a lot of mussels and crustaceans. An eel pout trying to sneak into the mussels there. High correlation we've seen with coal seeps and methane seeps and large amounts of diversity. Makes sense because a lot of the energy that powers our deep comes from these coal seeps.
So we finish here, then we'll start moving to this point, and this one, and this one. So we'll start at these points, and this one.
On the ridge, yeah?
Yeah, so there's this one, and the second.
So you want to come almost halfway between those 2 waypoints, and then down to the right.
Exactly.
This one.
Yep.
Yeah.
So
We will start like visiting all the red points.
So here,
So where's the red point relation to this? So this is where we go by. Yeah, we could watch this red point, but we're not sure.
Yeah,
Because
what that,
So,
what
so what we
Zach
know,
did
we have to go west.
is basically going through the edge of that map.
so you
So
can see
it's-
waypoint 3 on here, 3
Yeah.
and
This
4.
line.
So it's 4. You have to head right 4.
So we're going to come to about here.
Yeah. Yep, I guess so.
6 looks to be this one here.
6.
Yeah, no, the red points are where we find the bubbles.
Yeah,
in
the
map.
red points are on the waypoints.
And the
Yeah.
these are the waypoints.
Correct.
So it's the same one, but that one is missing the red points.
Okay. So we just following this line, what we have here.
No, we,
No.
Want,
More or
we want
less.
to follow. See where it's at.
We want to follow the edge.
So we want to follow
You want to follow this?
that edge, yeah,
Okay.
because that's where we have been finding.
Yeah.
So yeah, hitting all the red points basically.
Yeah.
But we don't want to get
just
down
wait.
here.
As we are.
After we get this
Yeah.
one, we will start going
Yeah.
down.
248, 250 roughly.
Yep, so we're just sorting out our direction. We have waypoints, but we've also, when we were doing our scanning, we picked up on some likely areas for coal seeps. So we want to check those out as we move along to our 4th waypoint. So as we continue to explore our deep sea, just a special shout out to my little nephew up in Hereford. Hi, hi, Louis, little Louis. I know you're peeled to the screen every day. Don't worry, Daddy's gonna be home soon. He's working hard. He's actually driving the ROV right now. Don't forget to put on your sunblock when you go to Spain. typical of areas close to
Yeah.
to seeps. We see a lot of shells, a lot of bivalve shells,
The message.
Is there a message on it?
and tube worms as well. Tube worms tend to, what I've seen, tube worms tend to point in the direction of seeps. I'm in the hot seat, and we have two of our ROV pilots driving the vessel. And have a good night, Louis, up in Hereford.
So every time you see one, you want to just
Take a look.
take a look.
Yeah.
There's bubbles below us.
So when we see any large collection of shells, we just want to take a quick scan, see what we see. Well, sea star, and we have some mats of bacteria. Always interested to see what's over the horizon. As we dip down a little lower on the escarpment, following likely sea points. They want us to go to this one first, and then cut back.
And I think we head into the.
Yeah, we are heading along to there.
We hit this one,
Yeah,
Yeah.
and
and
then
then
we want
this
to go
one.
to this
Yeah,
one.
so I think based on this, I think we are heading there.
Yeah,
Too
we are.
flexible,
Yeah,
that's
very
same.
cool.
Yeah, maintain. Yeah,
Yeah, yeah.
looking good.
You think it's possible to put that, those
Huh?
the red points in this one? Is it
Yeah,
possible?
it should be there.
should
You
be
guys
there.
weren't supposed to be putting up the
But this was, Juliana put them later, but I'm gonna ask her.
because it's.
No, but we okay. I think we heading in the right direction. It's just when we have to cut back.
I mean, I'm sure it is, but you're gonna just have to hand punch in the coordinates. I'm not sure if you have a list of them or what.
I mean, let's see. I was gonna. I mean, the problem is if you show them, we just leave it going between them because should we show we go between them? But we
So methane seeps are some of the most remarkable ecosystems on Earth.
Yep. Yep. Perfect. That's why I was gonna suggest.
It's also known as sometimes as a cold seep.
You'd have to have them copy.
It's called a cold seep because unlike our hydrothermal vents,
Yeah, that's a
the fluids that emerge from these cold seeps are close to the surrounding seawater temperature, whereas our hydrothermal vents tend to be much higher in temperature. These seeps exude quite a bit of different compounds, things like methane, hydrogen sulfide, some other hydrocarbons, and basically mineral-rich fluids. As we saw over this feature with lots of little tube worms.
Yeah, it's a bit off.
Oh, I see.
Look down close.
quite different to our abyssal plain that we explored a few days ago. We do have quite a bit of relief down here, which tends to promote a lot of growth as well. We're at the edge of this shallow canyon, and we're trying to explore the edge of it. And work our way down the edge of this relief. Some possible bacterial mats there. And a special message from one of our ROV pilots, Manny, a good friend of mine. Shout out to Charlotte, Louis, Otis, and Maya. Daddy boy's missing you all, and Spain's not too far away. Wish I could join you guys. So the chemicals coming out of these cold seeps, these methane seeps, provide the energy for an entire ecosystem. When you think about it, most of the life on planet Earth is actually powered by the sun, but in an environment thousands of meters below the surface, the sea surface, no light penetrates here. Instead of photosynthesis, these communities are powered by chemosynthesis. So you see a little halo saw there, possible halo saw, or rat-tailed fish.
Another one, another rockfish.
Quite a bit of diversity here, both live and dead mussels, tube worms, a host of crustaceans. Yep, our trajectory looks pretty good. So while we are trying to stay on a particular track, when we do see something interesting, we do tend to explore a little bit. So that's what we're doing. Quite a bit of diversity here. So we're taking a little bit of time just to see what we see. Looks like a sponge wedged into the rock. A lot of sedimentation on the rock, possibly calcareous. An anemone, a tube worm, once past this feature, I think we need to come to the right a little bit.
The water was across here.
Yeah. If you could come to the right and straighten up on that red point. We want to remember we want to hit this one first, huh? Cool. So back to chemosynthetic bacteria that use chemical compounds like our previously mentioned methane or hydrogen sulfide as their main energy source. They combine these chemicals with oxygen to produce organic matter, very similar to how our plants and our plankton at the surface use sunlight to produce carbohydrate energy. So these chemosynthetic bacteria form the foundations of our deep sea food webs. Without them, really and truly, none of the larger animals that we've seen would be able to survive here. Everything is in a very, very delicate balance. Yeah, perfect line. So we're ticking along at about 1 meter per second. Tube worms are quite long here. Shout out to Leah, one of our regulars. Happy to have you, Leah. We've seen quite a lot of tube worms. These are quite extraordinary animals. Many seep tube worms actually don't have mouths, stomachs, or even intestines. How do they obtain their energy, you may ask? Well, they do so through symbiotic relationships with millions, billions of chemosynthetic bacteria, which they house in specialized organs called trophosomes. The worms supply the bacteria with the necessary methane and hydrogen sulfide, which they obtain from the water, oxygen, and carbon dioxide as well. And these amazing bacteria convert these chemicals into carbohydrate energy / food and share it with the worm. The worm also provides protection for these bacteria.
So you also can see small tube ones. You can try to collect those,
Okay.
but not the bigger ones because
Yeah, that's tricky.
yes.
So if we do see smaller tube worms, more manageable sizes, we can try to collect. The big ones trying Trying to store those is be a real problem. And please keep your comments coming in the chat. We try to get all of them. Not going to be able to shout out everybody, but happy to have your questions and happy to have your interest in the deep sea. So we've come up on one of our interesting spots that we saw whilst mapping. You want to pan to the left a little bit? Are we okay? Okay. So, this one.
Oh,
This one?
Yeah, we will not get here.
Okay.
We will just like start doing one.
Okay.
very cool one.
Yeah, we gotta kind of turn to our left. Maybe 30 degrees. Something
We were there. This is- It's a jellyfish. We saw a jellyfish. We saw dive.
a type of jellyfish. Beautiful color as he wafts away in the current. Other types of organisms we see a lot of at these seeps are mussels and clams, who also have symbiotic relationships with bacteria that are housed in their gills. Many of the crustaceans you see around these seeps come for the food associated and produced by some of these dying tube worms and dying mussels. So we've just changed tack a little bit. We're going to explore another one of our likely spots for methane seeps. We're literally at the edge of the escarpment, so you'll see it angling down. Many of the things we see down here have never been recorded for Trinidad and Tobago. So we're breaking records every day. One question I see in the chat is what causes those lines in the sand? Most of the time, organisms create those lines, but those kind of snake-like patterns are often made by or sea cucumbers as they move along and siphon up the substrate, eating and depositing their scat as they go along. So sea cucumbers typically make those snake-like patterns in the substrate. Quite a bit of fishes as well, some halosaurs as we saw, yep. Yep, so we're right on top of what looked to be a likely site when we were doing our mapping.
If it's not there, we can continue to the next one.
Okay. Next one.
Yep.
You see it in the radar?
No, no, that's not how we do that.
Observation general.
And expanding our knowledge of the deep sea of Trinidad and Tobago would not have been possible without some of our partners and contributing institutions. I like to shout them out just to pay a little, give something back to these amazing institutions, these far-reaching, far-seeing institutions like our hosting organization, Schmidt Ocean Institute, and its flagship vessel, the research vessel Falkor 2. As many of you know, SOI is a private non-profit foundation dedicated to advancing oceanographic research and sharing knowledge of the ocean. And we're quite fortunate that they granted us use of their vessel. But in order to be successful in our bid, we had to go through quite a rigorous process, and that process was led by Species. Species is a non-profit NGO located in Trinidad and Tobago that undertakes scientific research, education, and advocacy. Many of my colleagues on the vessel are part of the Species organization. So kudos to them and shout out to them. Other institutions that have contributed significantly to getting the vessel here and to the research that's aboard the vessel currently, that's going on the vessel currently, are the University of the West Indies. Oh, it's an octopus. We're just gonna take some time here. Get some cool images. Wow. Majestic. Looks to be about 20 centimeters across. Unconcerned by ROV Sebastian. Stretching from his nap. interesting thing about octopi, they have pigments in their skin, chromatophores, that allow them to change their color and their pattern based on their mood, based on their environmental conditions. Just getting a little bit of turbulence and turbidity from the ROV props.
Yeah, let's stay here to get some good footage.
Yeah. So we're gonna just lime here a little bit, as we say in Trinidad parlance, to get some imagery of this majestic creature. Just waiting for the turbulence to settle. And a special shout out to the University of the West Indies, Faculty of Science and Technology, our Dean, Professor Adesh Ramsubag, and the Department of Life Sciences, and our Head of Department, Dr. Mike Otham, as well as my colleagues there at the Department of Life Sciences. You all have a good day. See you soon. Lasers on? Can you just put on the lasers? So hopefully, yep, we're seeing the lasers on the screen. You can get an idea of how large this individual is. And it helps us as well. I'd put him at about maybe 60 centimeters from tip of arm to tip of arm. About 10 centimeters between his eyes. Still a little turbid. We'll try to get some frame grabs here. With me on the logging station currently is Mr. Ryan Manette, and our dive lead is Dr. Bia Naranjo. Other contributing institutions: the Institute of Marine Affairs, Harbor Branch Oceanographic Institute, Florida Atlantic University out of the USA, the University of Calgary, Canada, Dalhousie University, Canada, National Museum of Natural Sciences, Spain. And as you might imagine, we have a quite diverse crew and a quite diverse science party. I think we have representatives of over 20 nations on board. So after getting some good imagery of that guy, we're going to move on to our next likely seep point, a little bit lower down on the escarpment. Other supporting institutions of the expedition are Ocean Discovery League USA, Pure Ocean France, the Explorers Club USA. Always brings a smile to my face when I read that name. The Pew Charitable Trust USA, and the Anthony N. Sabga Awards for Caribbean Excellence. Special shout out to these institutions. We would not have been able to do the expedition and do the interesting scientific work that we're doing without your able support. So thank you very much. And thanks to all of you who are tuning in to the channel. It really does help the channel and help to spread awareness about our deep sea and the conservation of our deep sea. So we're just reorienting. And heading down to our next likely site for methane seeps.
Maybe when sun shuts out
The octopus. So we're trying to get some last few shots of the octopus. It is a bit turbid here. As we get a different angle on him.
I mean, I don't know how they are in Trinidad waters, but
Just getting a little bit closer.
So I think current is moving at this side. So if I come back on the next side, current is not blowing away, but I think current is blowing. So the vest is going to be shitty. So I could land and try.
that's good. That's good.
So you may be hearing another Trinidadian accent. That's one of our ROV pilots who hails from Trinidad. So those of you who are thinking that this is a cool job, it is doable even for us Trinidadians.
Where is that?
We got as he sends out his arms to try to taste what we're up to. Constantly getting inputs and information from sensory organs located on the underside of his arms. And apologies to my brothers and sisters in Tobago. Yes, we are Trinidad and Tobago. There's no difference between us. We're all Trinbagonians, as I see in the chat. So big up Tobago as well. Wow, that's an amazing shot. Very large eyes. I wondered what are the things that he must have seen. gonna sample them, right? No, we're not taking it.
Yeah.
No, we're not gonna do it.
Gonna take it?
No. So all we'll be taking right now are pictures and some video. We're gonna leave him be. The camera work is amazing. As the turbulence has settled, we're getting quite a few lovely shots. Good. When you're ready. So we're gonna move on now to our next likely seep point. Hopefully we see some bubbles. That's always exciting, especially for geophysicists on board. Good heading. We're currently at a depth of 1,686 meters. It's a balmy 4.32 degrees Celsius. And we're on a heading of 100 and also 80 degrees. Sea cucumber. Difficult to tell the age, although things do take a long time to grow at these depths. Things are really slow. So he may have been around for a while. And I've just been informed that our beautiful octopus is a musso octopus, and it is actually our first record for Trinidad and Tobago. So once again, our expedition is breaking records and generating brand new information about the Trinidad and Tobago deep sea. Amazing. Possible Brisinged. Some brittle stars off to our right. Yeah, so ROV is serviced after every dive, and all of our biological containers or sampling containers are cleaned out. It's more to not carry over organisms from one dive to another. But yes, we do ensure that nothing is entering that should not be entering. And we're very specific to not leave anything unless it is for experimental purposes and to be removed at subsequent dives or subsequent expeditions. Some large sea cucumbers, brittle stars, and some bivalve shells. So we've just come up on our possible methane seep site as we cast around looking for anything likely. You want to go to the lower site or the upper site first?
No, we'll visit to this site, not these ones.
Okay. Well, I guess the closest one.
We'll confirm the other ones. Yeah, they have it here.
Cool.
We're going to 19.
We have it now.
Okay, cool. Perfect. Another sea cucumber track. So whilst doing our mapping earlier, we picked up on quite a few likely spots where there may be cold seeps. It seems to be a bit of a hot spot for cold seeps, so we're doing a little bit of exploring. We're not doing any surveys currently, just trying to find some likely spots where we would encounter some different diversity. Some pretty large crabs and tube worms.
ocean?
So we're just gonna cast around here, see if we can pick up on any bubbles.
You want this up there?
The smaller ones, if you could get. Smaller
This
two
is too big
rooms.
still. This is
Yeah,
good
that-
enough or no?
Yeah.
Yeah.
The same.
If you could get the smaller ones, yeah.
It's a super huge one, but the smaller ones might be a good fit for the biobox maybe.
So you wanna try that?
Yeah, and the containers.
Which I'm using money. How are you gonna? You said it breaks.
Yeah. Try to get it as close to the router, Manny. So oftentimes we react to what we see or react to what we see when we are mapping. We do, as my colleague is saying, we do have plans, but the nature of the exercise is one that can be quite opportunistic. If we see something that varies from the plan and it's interesting, we're probably going to explore it because it's never been explored before. It's a lovely example of a tube worm out of the tube. And some associates as well. So that guy looks to be maybe 20 centimeters. No, let's go for an individual. What about this guy right here with the associate? Doesn't seem to be too long.
So we just pull it from the stem, pull it.
I think it's longer than you think. I think it's like
On the other side?
Yeah, it's actually one of the biggest ones.
Well, maybe trace one from the root, Manny, and see.
This
Yeah.
big rock is in
I'm sure we
front of
will
us.
find a better place.
Okay.
Like
Okay.
not some
A boat?
there's
A
bigger
boat, yeah.
rocks.
All right, so we'll relocate, try to find some smaller worms. We don't want to take one of the larger ones because it's problematic to safely get them into our bio box. And with me being one of the people responsible for preserving them, it's really difficult to find containers that can fit the shape of these organisms. So we're going to aim for something a little smaller and continue on our exploration. So we'll get a better shot in a bit when we find more likely individuals. We'll take some time to get some frame grabs and some videos, as well as collect a representative sample of the organism. These samples will be housed in the UE Zoology Museum and the IMA's Marine Museum. So if you're visiting or you're even from Trinidad and Tobago and you want to see a close-up of these guys, you can come down to the UE or to the IMA and take a look at our deep sea displays. All tours at the UE are free, although we do encourage donations. We're open to schools as well as walk-ins, and any citizen of Trinidad and Tobago is welcome to come in and take a look. If you're a visitor from outside of Trinidad, just shoot us an email and we can set up an interesting day for you. So come on down to the UEZM and the IMA's museum. Send us an email and we can work out a day that works for both of us. We're open Monday to Friday, 9 a.m. to 4 p.m. Also housed at the Department of Life Sciences is the National Herbarium of Trinidad and Tobago, where we keep type specimens and representative samples of all of the plants that you would find in Trinidad. If ever you want to find out what something is, you can go and check out its curator, Dr. Juan Francisco Morales, and his amazing team at the National Herbarium. As we're joined by Professor Anna Metaxas, who's taken over dive lead. And Dr. Rachel Lauer, one of our resident geophysicists.
Go to dinner, bud. I'm here.
So thank you all very much for the last hour of your time. I'll now pass over to Dr. Lauer, one of our, as I said, resident experts on deep seeps and cold seeps. So, from the midnight zone to your screen, thanks for diving deep with the cucumbers and corals today. This is Zahir signing off from the deep sea, where the pressure is high, but the thrill of discovery is even higher. Thank you all.
Thanks, man.
Okay. Okay. Cool. Do we know which the numbers for these guys?
I didn't write it down, but you can write it.
Okay. The numbers for the Niskins.
377. 378. 378.
Okay. Cool. Thank you.
No, no. Blood muscles,
Oh, there you go.
Thank you.
Hmm. Dinner tonight is really good, so make sure. So what are we sampling, if anything? I think, no. But they were having trouble getting them. They were asking for littler ones, if possible.
Yeah.
Yeah.
So if you say
Okay. Yeah. Can we see the heat flow probe? So, oh, there it is. All right, we're good. All right, good evening. We are heading to another site where a seep was identified in the geophysics data. And so far, this site has been lots of patches of mussels and some of them enormous patches of mussels. And we're just coming to revisit a few sites that we did not get to earlier in the dive and that showed up in a couple of the mapping passes that we did both earlier today and last week when we first mapped this site. I have a question about sanitizing and ensuring that things don't get brought down to the bottom that shouldn't be there or vice versa. That is a huge consideration for us, especially for those of us who are sampling for eDNA. It's obviously a big deal for NASA because they've had some issues where a lot of things that ended up in space that were from Earth, and obviously that's not helpful in the search for life. But here, yeah, we are very cognizant of ensuring that we're not bringing anything here or bringing anything back or contaminating anything that we are trying to preserve or sample. So yeah, it's an interesting question. Looks like another mussel bed maybe up ahead. A small one, no, dead one maybe. Yeah, some sargassum, not mussels. The insides of their shells really pick up light and are helpful in trying to identify some of these patches. Okay, there's some definitely living mussel beds and tube worms. Huh, yeah. Cool, yeah. Small, but there. Sure. I don't know how hard it is to sample from.
What's that?
Which ones do we want? The tube worms or
Yeah,
the,
that's what I mean. There
Oh,
some
were they
smaller
small?
ones,
Yeah, because we tried to take tube worms, but I don't know if you saw they had to like break them into pieces and fold them, and they were kind of. Yeah, you were okay.
That was
sorry.
it.
I mean, no, I'm not
You
saying
know,
there was nothing wrong with that. It was just like really hard to get them in the
Yeah.
bio box. So we were thinking if we got a smaller, intact full one, we could not have to.
Or 6.
Or 6? Oh my god. Okay.
David says you was 8.
Oh, 8 now.
We
Wow,
have
the number went up. Okay, cool. Yeah, because those really long ones, you know how they were hard to. You had to break them to get them in the bio box.
Yeah, those are babies.
These are
They're the
babies.
same kind.
I think so.
Okay.
Just babies. Now the one that's sticking up is a different kind.
Okay.
Yeah. She wants those two. Another 6.
The one that's sticking up, you want me to stronger?
Sure.
Yeah, sure.
I might have to just move a few of those muscles around. That's okay.
Yeah, yeah, that's fine. Does she want some of those mussels too?
So I usually
The boomerangs?
put them or just
That's what the others. I thought they got muscles.
Yes, so I'll
You got
grab
some
the
muscles.
scoop.
They have muscles.
Come in under the end of the boot.
Okay.
Some big boomerangs. Ooh. Big
Those are. I don't think she meant she wanted them. I think she was just pointing out that they are large. I think, right?
Right.
Yeah. Oh, there's another little eel pout.
Bread chart, can you pause the move, please?
So the. All right, so we have some smaller patches of living mussels here in these smaller beds, and we'd like to take sample of some of the tube worms, and ideally getting it from the base of the tube worm so it remains intact. Right? I mean, Ugh. It's been a long day, guys.
Can we take some video first so that we could show her the worms? Maybe.
Yeah, before we
So if we can
before we
just-
scoop,
before
get
we-
some footage. Sure. I assume the one you were interested in was the tall
Well, there's also the small ones, right? So
or the
what-
small one off to
Yeah,
the
'cause what's
side.
important is that we actually have intact tube worms rather than being gigantic.
Yeah.
So there's a whole bunch of small ones over there that would
Under
fit. Yeah,
the
the mussels.
can we
yeah,
can we
or,
move the muscles out of the way and yeah.
You know those ones.
Yep. Okay, but if you want to get them by the base,
Yeah,
we'd
all
have
the way.
to.
So,
Okay.
You know, you can
Okay.
see the root in that huge one that you took the first time, that black root.
Yeah.
We want to have that.
Okay, so we're gonna move them out, the mussels out of the way, so we can get access to them.
Maybe we'll see those.
Maybe we will. Yeah, so that little fish that's in there is an eel pout. One that just left. Oh, wow. It's a good way to see what's in there.
It can go pretty far back.
Yeah, it looks pretty exposed, I think. Is that, how long is the root? Is
It's-
it?
It could be this long.
Oh,
so
it
they
could
may
be.
dip into the
Okay.
sediment.
Yep. Somebody earlier asked if we could eat these mussels. I would definitely not. They smell like sulfur. They're really stinky when they come up here because they've been sitting on a vent.
Back around to
Did you want more food put aside?
More food. Yeah, they have the pilau,
Did you want more food put
like a
aside?
scoop of that. It's really, really good. I inhaled some, but
Oh, what?
pilau,
Halal?
Palau, I think.
Yeah, Rachel, that rock hard,
Is it
rock hard,
the base of it?
the base. This is me pushing down.
Oh wow,
Oh, is it hard?
That's it.
wow.
Not go down.
Okay.
Okay, well, we want them intact, so.
Yeah. Let's see. That's wild.
Okay, now they're all in there.
So that's like carbonate there, like rock. Okay.
If we can't get them, then just
Yeah.
move on. We don't want them broken up.
Yep. Okay. We'll find other ones. Thank you for trying.
So the woods of place.
What's that?
There is a waypoint to put in, which is 2 grams over that.
Okay.
So there
That's gonna
was,
move
so
on.
maybe, and it's that way, so.
Back that way. Okay. Cool.
I'll just hold this for a while, just 'cause we still.
Someone had a question about the chemistry sampling. Yeah, we have seawater on the ship, like taps that are taking in seawater. All of the samples that come in are, basically not being contaminated by anything else. And a lot of the chemistry sampling is done from either the pore water or the sediment itself. So I don't do chemistry, but there's a lot of protocols to ensure that that you don't have any cross-contamination from anything that we have on the ship. Yeah.
After this area of interest on the 19th,
Yeah.
where do you want us to go after this?
To
So what I was saying was beer.
is what zigzag between those.
And then go here.
Yeah. Is that cool?
Yeah, it's fine.
Okay.
Perfect. So we're gonna zigzag all of these cords.
Yeah. Oh, you put them in. Cool. Yep.
Yeah.
Go.
Awesome. Thank you. Alex, I assume you did that? No? Who did that? Magic?
Hero.
Alex and Juliana.
Juliana.
Join them.
Okay.
Which are being waypoints updated due east.
Up to the waypoint.
So when you guys go, before you go down, you put water in the little quivers, right?
Yes.
Yeah. Is it like warm water, or is whenever they shimmer when it opens up? Is that? Is it just
Just
fresh
fresh
water? Okay, so just fresh water. Got it. Oh, cool. Oh, another one. Cool. Is that on our, not quite to our point yet?
Not quite, not far away.
Yeah, looks like it keeps going. Nice. I got it, yeah. Wow. Okay. This one's also pretty extensive. Wow. All right. Yeah, let's take a minute to look around here. This is wild. Cool. And how far are we from that next waypoint?
This one?
Yeah.
20 meters.
How many?
20?
Oh, that's it?
Yeah,
20 meters? Okay.
20 meters.
Okay, so that's probably this. Nice.
Looking at the sonar, this goes on for,
Yeah,
this is all around us.
cool. So we can
All
maybe.
of the white is rock.
Wow.
All of this is
Okay.
maps.
Yeah, maybe we can.
Let's look here.
Oh, look at those tubers.
Yep. Do you want to first come up and look
Oh, yeah,
around?
drop it,
Yeah. Maybe we can just come up
size
and
it, yeah.
see how big it is, so we
Yeah, for
know
sure.
the area of the seep, and then we can maybe come back for tube worms if it's like.
I haven't tried the fish.
Good.
Good to see that. Such good photos of
Fly
If they're in the rock. Yeah, like through the rock.
So you come here. Good. You just want to overview.
in front of us.
I just see where we just came here, so that's the see.
Thank you.
Yeah.
Look, you can draw like those smaller. You can draw any color, you're not gonna fit in the map.
Say that again.
You can't draw any color, you're
Oh,
not
yeah.
gonna fit in the map. You have to fit. That's it. Stop drawing.
So once we can see again, we can come up. Just we want to see how big this is because we know it's a mussel bed, but we haven't really seen how big it is. Bless you. And we want to eventually head to that waypoint once we get like an overview of how big this is.
The sonar seeing rock.
Everywhere, huh?
100 meters out, as far as the sonar can see.
Okay.
It's very flat. So it's
Yeah, there's these slabs that carbonate that are slumping down the hill. Lots of tube worms.
So you see, we have been not here. See, Rachel, there is a waypoint.
Yep.
On
On the
Tobago,
east.
just off there.
Yep.
So I think those were the ones that were in the sediment, were they?
Uh,
No, it says mussels.
It's labeled small mussels.
Yeah.
yeah. So not far from the where we put it.
Between those, yeah. So I suspect this is pretty extensive. Like not just this, but the next waypoint.
So you want to head up to 19 and 16?
Yeah.
Sort of
Yeah.
patch.
Yep.
A patch, and then
Exactly.
it
Yeah,
goes
kind of
and
up there and then
patch.
zigzag down again. But we can check that one off as another seep, whatever that number is. Chocolate?
These waypoints here.
No, for Rachel.
No, no more chocolate for me. I'm
For
cut off.
Rachel. She's cut off. You can only have chamomile. Nothing else. Sleepy tea. That's it.
Yeah, that's just to see how big it is. But yeah, this is good.
Bridge on it,
And we can head towards that
Well, take the waypoint,
waypoint, I guess. So do you want to sample them here?
Look, there's lots. They're not
Yeah.
that tough to work.
Okay, we can. Are those look big though? They look pretty big, right? These are too big. Yeah.
Yeah, let's just move on.
We'll move on. If they're too big, we have to fold them to put them in the box, and then
Thank you.
It's not good for the tube worm. Oh, look at that. More over there. Wow. Just keeps going. What's that?
All this.
Yeah. Yeah, she sent a little dive plan to extend it. Yeah, there's just a lot of stuff in the water here too. Mhm. So just a recap on what we're doing. We have been over this area twice with multi-beam and identified, I don't know how many potential seep sites that show up as anomalies in the data that look like something coming off the bed that is likely bubbles of methane, and those tend to correspond to these mussel beds. And we're just in the process of visiting as many as we can on this particular site before we surface again, and collecting some samples along the way. We've gotten some mussels, some tube worms, some more tube worms, and I think there's another species of tube worms that we're trying to collect as well before we surface. They're just everywhere. They're everywhere. So what about the smaller, like we were saying that the larger tube worms are hard to, because you have to fold them to put them in the bio boxes.
Yes, I did
And these are huge, right? These are too big.
Yeah, it's complicated because even when they seem small,
Right.
they are like
Really brittle.
under the ground.
Under the ground,
They're
okay.
always bigger than we thought.
Okay.
Did you get any?
No,
No.
no, we tried earlier, but they were behind rocks and we couldn't get them out. So we're still looking around for good places to get some. And also the right size, right? Not the, nothing too big.
Difficult tasks.
I know,
for,
yeah. For a biologist, for a geophysicist, yeah.
What is the thing with the tube worm?
What is it?
Yeah, what is the thing with the tube worm?
Why do people want to study them? Well, I think one of the,
Well,
they
need
take a
some-
long time to grow. They are an indicator of, or concentrating pollutants potentially, right?
Yeah.
Maybe microplastics. Um, I don't know.
Yeah, and they also need for vouchers. I think
Yeah, and they're also representative species of what's here. Yeah. I personally don't study them, but. Oh, here's another. Cool. Yeah, it's like, I mean, this whole area is just covered in mussel beds that are patchy,
Or
but
they'll be higher here, then they'll go back down.
yeah, sounds perfect. Yeah. Bubbles? Where?
You're there. Those
Here?
center.
Oh, I see. Okay. Oh, yeah, yeah. They're burping out from that thing.
Square.
Yeah, right here. Lots of bubbles. Oh, down here, I guess. Nope, further down. Okay, right here. Cool. We have bubbles. Yeah. Okay, let's hang out for a minute. This is mesmerizing. Yahoo! Bubbles. All right, I'm sending a text. Wow. Okay. So it's coming out from this mussel bed, it looks like. But there's a lot of bubbles happening here. So that's methane. Ooh, nice burp just came out from under that rock. Yeah, it's like all down here. Cool. Wow, it's just, and it's coming out from up there. It's coming out of everywhere. And we have the usual suspects. Got the shrimp. We got some, oh, the mussels. An eel pout is sticking its head out from behind the leg of the sea star. Oh, look. Oh, I have a soft spot for these guys. Oh, wait, he's being attacked by a shrimp. Wow, nice shot. We don't want this, any of these, right? We're just,
I would say maybe the sea star because we haven't grabbed one.
yeah,
And we've seen like a bunch of them.
yeah.
Yeah, let's go for it.
Okay, we have seen a bunch of these sea stars, and sounds like we're going to take a sample. It's quite a community, yeah. So
Sea star?
that sea star is ours. Or yours.
Good.
Those are like cool. This one.
Yeah, but I didn't see it
It's
on there.
nothing
No.
cool, which is a good sign.
Right. Maybe it's
It means-
new?
I mean, I don't know.
know.
Like, if it's not here, it's like, it's not,
Okay.
yeah. So,
Okay. He's also right on top of that seat.
yeah.
He likes methane.
It's gonna smell good.
Will be a stinky sea star. Wow.
Sorry?
Look at the, yeah, there is just a ton coming out here. Like last week we saw one bubble, we were like, woohoo! Now it's like,
Ordinary.
yeah, like, yeah. Cool. No, but this is a great site. We have all the cool animals. Oh, the eel pout is hiding behind the star. Maybe defending the star. What's he doing? Oh. We've seen like 5 or 6 of these sea stars on today's dive, and oh, gotta zoom in on there before we sample. I guess you kind of already got one, but maybe another one
Yeah,
just
this
for,
one is better.
yeah. Got it. Okay. Oh, great question. So, yeah, the methane that is bubbling out here, we're at what, 1,700 meters water depth, and this methane is coming from probably kilometers below. Well, it definitely kilometers below. In the, sort of the hydrocarbon source region. And I'm getting text from Anna about, she's saying use suction. Yeah.
It's so small that it's probably
We can also try that. So I'm just afraid that we can.
Yeah, so the, we're just figuring out the best way to get this sample. Yeah, but a lot of that methane is actually metabolized by bacteria, and along with the hydrogen sulfide, and is ultimately metabolized into the rock that we're seeing here. So it's sort of, that rock is calcium carbonate, and it is biochemically produced in these vents and cold seeps by critters that are metabolizing that methane. At these depths, that methane is probably not making its way to the surface. That is a concern when you're in shallower depths, for sure. In the deep, there's a lot of critters that can metabolize that methane and use that for energy. There's not a lot of sources of energy down here. This is probably the first time any of these critters have seen light, so. So depending, I think this will, is the plan to,
Ideal going.
Yeah.
It's so small.
okay.
Cool.
Nice one.
Whoa. Wow.
Lots of biomass.
Cool.
We have any of the fun ones.
Yeah. BB,
Open the punch now.
3B, up to you, whatever's easiest, I guess. But you've got 4A and 3B in the front.
Which one was the first one?
4A? What number are we on, Rob?
This is 379. Where's it going?
BB.
BB 4A.
Yep. Oh,
Yay, well done. All right, so instead of using the the arm and the claw on that one, we used the suction sampler to move the star delicately away from where it was sitting and put it in one of our bio boxes. I know. This is just, this may be one of the best seeps we've seen in terms of volume that's coming out. Our site yesterday was, you know, most of this methane is trapped in the seabed. There's a few ways you can destabilize it and have it come out. Yesterday we were on a landslide, a submarine landslide that looks like it exposed methane beneath the surface, and that turned into a seep. Here it looks like it's bubbling right out of the seabed. And
shrimp
pretty,
like it, right?
yeah, and these are the chemosynthetic shrimp that really thrive on methane, and the tube worms and the mussels. Yeah, it's not clear how much of that is, you know, getting consumed here versus going up, but obviously methane in the atmosphere is something we want to avoid for obvious reasons. Oh, look at this little guy down here too. Cool. Do we want to eat these sponges? No? Yeah? All right. Do you want to point which? I can't tell which of these, yeah. We have a sponge expert in the room who would like a sample. Down here?
Yeah,
This guy?
Yeah.
Okay.
That one.
Yeah.
Boop. That. All right, right above the crab and below the shrimp.
Suck it?
Suction? Or too big? Maybe cut in suction? Or
Yeah, it's very small.
before we do that, we should take a Niskin, right? Okay. All right, so. Does it matter which Niskin? Just go in order? 3? Okay.
Just one.
Yeah. That'll be 380? Yeah? Okay.
Yep.
What? This is a starboard 3. Starboard 3. Oh man, what's going on? I'm so slow today. Okay, going over.
Oh, they go? Okay. That was 380?
This used to be with the arm.
What?
Before?
Yeah,
for
they used to
biscuits,
do it was
yeah,
with
they
the
have to
arm.
put the arm.
Yeah, with the tiny little, yeah.
So that's not new.
No, this is better.
Progress.
Progress, yes. Yeah, I remember. You had to like, bing, bing.
Much easier, for sure.
Yeah. So while we're here in this awesome seep, we're also going to take a sponge sample.
The sponge, I think this is your.
Oh, it's the one.
Yeah, right there.
No, the one with the green dot.
The
Right?
green dot? Yeah. Is it hard?
Maybe it's in the rock.
It might be. What about the tube worm?
This is so big.
Is that a tube? Is that
Yeah, we can try that one.
good?
Is it too hard? That one. But no, sorry. I think we need to take them.
Oh yeah, we
That was
need
for
to.
later, yeah, sorry.
Yeah, we need. Sorry, we need that one. We need to get to the bottom of, unfortunately, because they have like a root, and the
Yeah,
there's a worm in that thing, and we need the whole worm. Sorry.
He was referring to the sponge.
Oh, right. That's okay. I mean, but that's a good sized tube worm for us to take, is if there's one in
Yeah.
there.
Maybe let's try the sponge that is upper, like this piece here.
Next to the muscle, or tucked in next to the muscle.
No, it's too.
Maybe it's too attached to the rock. Okay.
Well, there you got a piece.
Yeah.
Yeah. You will, maybe.
Where did that end up?
It's
I think it got
somewhere
it went up
away,
in the suction.
But it's on the tube still, I think.
Is it?
Yeah.
Okay. It's not in the. Oh, okay.
Yeah, don't log it.
Don't log it.
Not for now. I mean, not yet. No, let's go for the.
Hmm.
No, we just started. We'll go to
Do we
try
want-
for the tube worm,
For the tube worm.
but with the arm,
Yeah, we'd need the you know,
the base.
the claw. Yes. Yeah, so to some of the questions about methane seeps, this process of, it's called anaerobic oxidation of methane, which is this process that transforms methane into solid carbonate rock, which is a lot of the rocks that we're seeing here. All the rocks that we're seeing here are carbonates. And that process is basically locking away carbon on the seafloor for millions of years, which is a natural way to sequester carbon. And it's basically a partnership between microscopic organisms who are symbiotically working together with sulfate-reducing bacteria. And that process is producing, the byproducts of that is precipitation of calcium carbonate. So yeah, there's a ton of that at this particular site. And that's a process that takes millions of years to form this much carbonate. And that's basically taking that carbon out of the carbon cycle while it's stored semi-permanently at the seafloor. So while there is obviously methane being released, a lot of that is being metabolized and used to form these platforms, which are also providing hard places for other animals to live. So one of the things we're trying to do now is collect one of these tube worms, or more than one. And yeah, sponges are very, very cool because they are basically filtering tons of water. And for us, and also provide some of the cures for diseases that, I mean, they're some of the oldest creatures on Earth. I think they're 500 million years old in terms of how long they've been around. And over that time, once we started being able to harvest them from the seafloor, we've learned a lot about cures for diseases in humans that are found by studying sponges. So sponges are holding the answers to a lot of questions that we have in medicine, and there's a lot of medical research and biomedical research and pharmaceutical research that is focused on sponges. So very cool. And the cool thing is, if you're studying, like, if you want to sample DNA of other animals using a technique called eDNA, which is being used by one of the scientists on this ship, sponges are the things you target because they filter so much water that they are likely basically capturing and storing a lot of the DNA from the water that they're filtering. And so sampling them allows us to sample anything that interacted with that water that they have filtered. So they are basically, sponges are sponging DNA. Oh yeah. All right. I am gonna hand it off. We are in the middle of trying to get one of these perfectly sized tube worms that will fit into either the quiver or the bio box. The problem is you have to get them at the base.
Right.
And the other ones that we've seen everywhere are massive, right? And so if you get a ginormous one, you have to like put it into its square, and then you're breaking it, and that's kind of ruining the sampling.
Right.
So we're trying to get one that
Size
is,
one.
yeah, right, the perfect sized one. And then also, oh, cool.
Perfect.
Well done. Nice job. And we're on this awesome seep that has like all the things that we want to sample. So
Plenty of bubbles.
so much, yes. And if you would like to continue talking about the awesomeness of sponges, I've set you up. So there you go. Okay. Oh yeah, sorry, I didn't mean to. Should we put it with the other tube worms, or we put it by himself?
Let's use this one.
2B? Okay. Let me just wrap this up and then I'll, yeah.
This has sponge on it, so
What's that?
it has a sponge on it.
Does it? Okay.
It's covered by sponge, I think.
Oh yeah, yeah.
Might be dead. I'm not sure if it's alive, but
It's hard to say. Would it still be in there? I guess, is that still useful for DNA or no?
I don't
No.
know.
Okay.
Probably.
Is it enough? Look.
But really has a sponge as well. So. Some sponge for you. Some sponge for
Okay, so BB2A.
B.
B, sorry.
Yeah,
Yep, B.
That'll be sample number 382.
382. Wait, where's 381? I have 380 on Niskin.
381, he has a shrimp.
Oh yeah, it's fine.
Oh, this section.
Yeah,
Yep,
yeah,
yep,
section
sorry.
times
Yep. 381. Chemosynthetic shrimp. And 382. Which kind of tube worm is this one? I'll just put tube worm. Everyone knows. Yeah. All right. Awesome. Well, I look forward to hanging out with you all again tomorrow. And I'm not stealing your pen this time. Here you go. Yeah. I'm leaving you in the capable hands of Kyle, who is a real biologist.
Good, how are you? Okay, I think we're going to continue to try to make our way to waypoint 4. So just...
No, it's
Oh, is that not 4? Oh no, sorry. This one over here.
16.
16, right?
Yeah, 16. No.
16.
We are here. This is 16. We
Right.
have it on the map. Then we go to this one, which is
14.
14.
And then 9. Still one's in line.
Yeah.
Sorry.
And yeah, we will try to get
Go
to the top of this.
there. Yeah. Like,
So we just fly around those ones, see what is there.
Alright.
Okay, do you guys want me to send a few pictures of the collection?
You have some.
I can send 500.
Okay, hello everyone. My name is Kyle Foster, and I'm a postgraduate student at the University of the West Indies St. Augustine campus, studying deep sea biology. And today we are exploring some methane seep sites. We would have been conducting seafloor mapping using a multi-beam echo sounder throughout this expedition. We would have passed over the site a bit earlier, and we would have seen quite prolific bubbling from the seafloor. So these seeps have not been explored yet. We came to visually confirm the bubbling, and thankfully we have now come across a good few set of bubbles, so we're quite excited about that. And now we're just continuing our exploration, sampling where we can. And yeah, just keeping our eyes peeled for more bubbles. So throughout this area, would have seen quite a lot of tube worms. You would have seen we just collected one. Lots of shrimp and mussels as well. Amphipods, and pretty much all of the organisms that you see here are well adapted to make use of the methane that's being emitted from the seafloor. So just like how plants use sunlight to create energy, many of the organisms here are able to, through the collaboration with bacteria, are able to turn this energy, these chemicals into energy. So that's how these ecosystems function. Got a nice sea star there. And then I guess discoloring that you would see on the seafloor, the dark and light patches are bacterial mats. So that's the particular bacteria that is able to transform the chemicals into energy. Got lots of shrimp. And then if you want to understand the scale or size of what we're seeing, we've got two green dots. Those are laser projections that are spaced 10 centimeters apart. So you can basically estimate what we're looking at, the size of what we're looking at here. We'll also be getting some sediment cores towards the end of this dive to see what's, basically living within the sediment. So we've got loads of dead mussel shells here. And old tubes from the tube worms. And of course, with every site that we've explored so far, scattered, we've got sargassum seaweed on the seafloor.
We are on the 0.16. Is it gonna head to 14?
Yeah.
What's that direction? I think we should stay another 12 hours. It's too interesting. This type.
I mean, many of us would agree, but
Best lead so far.
you think so? I think
so.
yesterday
Nice
and
being
today
here.
has been quite active, yeah.
It's nice.
Seen quite a lot. That's because, well, thankfully we've seen a lot of bubbling as well. So,
I like this shell fields.
Mhm.
They're amazing. Like colors and the more life.
So we are
Yeah.
trying to
How they
confirm
are.
the
They're
sites.
massive.
Yeah.
Look.
So just to keep notes there.
yeah, it's pretty exciting to see. So we've got like some small pieces of a small piece of coral there, I think. Well, we've also seen quite a lot of skates today in and around,
I've seen that one kind
seep
of
area.
almost gray, whitish.
Yeah, like almost white.
Yeah, the eye was different.
Mm.
It's a very different eye.
Uh-huh. So I'm seeing a bit of discussion about hydrothermal vents and methane seeps. They're two very different environments. Hydrothermal vents, you get very, very hot temperatures being spewed out from the seafloor, whereas these are considered cold seeps. So temperatures are much different. Got a good few live tube worms here. Some squat lobsters and shrimp. And so this expedition is very important for Trinidad and Tobago. My colleague Jamie Lee Luchin, who's actually logging for us right now, she would have published a paper recently about the state of knowledge of Trinidad and Tobago's deep sea biodiversity. Within that study, they would have pulled together about 451 species that were known, and they're about, based on estimations of species and family richness, about 1,600 that are estimated to exist within Trinidad's waters. So there's still about 70% more that we just don't know. So it's important that we come down to these areas and visually observe and collect samples and expand what is known about Trinidad's waters. Got large bacterial mats in these areas. And I would have mentioned the sargassum seaweed earlier. This is something that has really been become a lot more, let's say, prolific in the Caribbean since 2011. So it's really been like inundating our beaches, and I've always wondered exactly, how it would impact the deep sea once it floats down, or how it, yeah. And plenty of people have also floated the idea of, sinking sargassum seaweed and using that as a method of carbon sequestration. So studies have been done to try to see what colonizes the sargassum in the deep sea. We've been making our notes as well. Sometimes, it just lays there, but other times we've seen it filled with amphipods, and then, other bigger species are coming around to maybe feed on the amphipods. So it's all been very interesting observing. So compared to previous dives where it would have just been, sand without any presence of methane seeps, we would have seen a lot of sea cucumbers, mainly sea cucumbers, really. So you can really see the change in diversity as we are around these seeps.
Sorry.
Yeah.
Can we see that sponge that is under the rock?
Over here?
Yes.
Okay.
Lower rounded, right?
This here?
Yes.
Oh, yeah.
Is that a sponge?
I think that is a sponge. Let's
Just
take a
covered
look.
in. There's something.
Não. Não. Fala um pouco cá pra mim, por favor.
Yeah, it might be that, but maybe let's just zoom in for a while.
Yeah. Can we zoom in a little bit more, please? Okay.
No, it's the rock carbonates, definitely falls, maybe something like this.
So we're just trying to get a better visual of this. And most likely, it does kind of look like a sponge that's just covered in sediment.
I think it's alive.
Photos with the lasers on and lasers off, and then we'll try to collect some.
Right.
Niskin.
Lures, não é? Como? Distinction.
Number 4? Okay.
Strength is lower.
So we have to like load up and charge up, and then
No, we reduce the.
yeah, shoot that. What number is that?
383.
83?
We reduce the speed because it can go like all of them at once.
Oh, okay.
Very low speed.
Okay, so we'll just get some photos now. And just some frame grounds.
With the laser and without.
Yeah, with and without.
I mean, yeah.
Got it?
Yeah.
And then without?
I mean, I said plenty without.
Oh,
when
okay.
looking at it.
So we can just
Good.
collect that. Yeah. Thanks. Alright, so do we want to just get the whole thing or just piece?
No, let's try a piece.
Okay.
And see what happens. And then we zoom in the covered part. So that will be okay.
Okay.
Where are we gonna place it?
If it's a piece, I think can be a chloride quiver.
Here? Oh,
But if
a quiver?
not, we have. I mean, we will not collect many, so it's, yeah, that one is okay.
Okay. So, firebox 3B.
Place the cabin for a quiver into the pillars. No bottom of the box. Leave the coral quiver open. And it fits with the moving.
dots on the
Oh, I have no idea.
Oh yes, now there's it.
The yellow?
Yeah, you see some.
Yeah,
Mhm.
you should zoom in afterwards.
How long does it take for a sponge to grow this size?
Good question.
Take a question. Yeah.
Oh, can we zoom in on it, please?
Updated, yeah. You know how fast do they recover? I mean, grow.
Yeah.
Just- They're not, are not
And then just a
static,
little
it's dynamic.
focus. Yeah, that's good. We can just zoom in and focus a bit. Then Jimmy got her pictures.
Like a
And
nugget.
yeah.
Cool.
Yeah. And then we can put it in.
Yeah.
I think we're gonna go in.
And then we should turn around to see the other side.
Oh yeah, we could turn the clock. Hmm. Good?
More sponge?
Okay, cool. Alright, that's good, thank you.
It was easy to grab this with this, right?
Yeah.
So you might grab a bit more after
Okay.
you put this in the quiver, please.
And put it in the same quiver?
Yeah, same one.
That was good. Shrimp on it.
Oh, there.
Yeah,
fast.
makes good.
Yeah, interesting. Sorry, Shrimpy. That's good. Let's get more photos of this one as well.
What?
Just let me know if you yeah. Oh. Yeah. Good. Alright. Yeah, we'll turn around. Okay. Good. Thank you very much. And And now it's 384.
It's like the littoral strip of one.
Right.
I have no idea.
745. Did you want to zoom back into the sponge itself?
I think it's like
Under.
can't see. Yeah, it's like maybe we should just zoom in there.
The yellow bits.
Yeah.
Yeah.
What I can say is that maybe something fell from the other things.
Okay.
Or whatever.
Okay. Perfect. Thank you. I think
Right.
before we move off, we just want to zoom in on the little yellow things to see what they might be.
Close there.
Unless there's something else.
Get more? The sponge?
No, let's close that.
Megan, did you want more of this one? Are you good? No, we're good. Okay.
Yes.
Go for it. That's it. Do we gonna take a picture of the
Yeah.
part the sponge was cut or just yellow dots?
Yeah, let's zoom in.
See what the
but
shrimp
I'm
are
not
doing.
sure how it looks like.
Ah, it's counting down.
Yeah.
Better
It was up earlier,
before,
yeah. Oh, I mean, it's interesting to see the shrimp come in where it's been.
Yeah, they're fast.
Yeah.
It doesn't help much, right?
Yeah, it's too sedimental.
Hmm.
Wow.
It's a nice color of the shrimp. Different.
Yeah, we took some of those earlier.
Are those different from the other, like,
Got
regular
things that
shrimp ones?
maybe.
Yeah?
That's what they got things that maybe.
Because the other ones are like
Let's-
ghost white.
Let's zoom in the
On the yellow one? Oh, we're gonna look
Oh,
at the
yes,
yellow.
they have more zoom too. Yeah.
Oh, that's good. It's just, yeah, it's so good. The zoom is immaculate.
What catches me is the colors, like
Mm.
amazing colors.
Yeah. And these things are tiny.
Yeah.
Yeah. Okay, we'll zoom in on the yellow
Yep.
the yellow bits now, yeah. But that was a good shot there.
Yellow. We saw some yellow bits here.
It's another sponge.
Yeah.
Yeah.
You just set it up for him.
Oh wow. I think there was a bigger piece. Can you zoom out a bit?
Yes,
Yeah.
I think there's a rounded one.
They're so cool. There's a ton of them. What's all of they? The yellow?
The yellow
Yeah,
sponges.
right?
Just yellow sponge. Very nice color.
Yeah.
Ah, it's in a shell.
Some kind of impurity in the carbonate, but that's what turns it.
That's the maximum, right?
There's a sponge on a sponge. That's the plan.
Damn it.
It's a stone.
It's a shell, clown shell.
Interesting though, it's like yellow and almost spiky.
Yes, it is, right? They're soft. Soft. You need to go closer. I can go a little bit ahead.
Are you interested in this? Are you interested? Okay, so maybe let's get a bit closer. See what we can do.
Might need to take the shell and the, well, if we do collect.
That's right.
Oh, look at the colors on that rock. So, in Barbados, because we're exploring the Barbados accretionary prism, there's a part of Barbados which is known as the Scotland District. And you can see basically these types of rocks there, and they're just absolutely beautiful, the colors. But of course, we are in Trinidad and Tobago's waters, but the Barbados accretionary prism extends throughout many Caribbean island waters. And Barbados is basically just the aerially exposed bit of this large feature.
So I think after this, just one we choose this one, and then we will go to the next point.
Okay. So, surely satisfied with?
Yes, but it will be hard to grab it because it's attached.
Yeah.
It will be like super tight.
Yeah.
So we remain for the future. Okay, so yeah, let's go.
We can move on now.
Thank you.
Yeah, thanks. Thanks a lot.
Sorry, can you zoom in on the floor for a bit just to see if there's a piece of rock that is loose? We can get it.
I'll take a piece of rock.
Yeah,
Like
no,
that
with
one?
yeah, not necessarily with a sponge, but just a piece of this structure to understand it better.
Mm.
Yeah, any of those pieces.
The
Yeah, that one looks with the bigger one.
You want to use the?
I think you can grab one of those pieces in the seabed. Brownish section or
I'll be there for that.
section?
Club.
Yeah.
I'm gonna have to bring back this up, but you're gonna have to space to just see what we're gonna collect. Put my apple in it. Tava solto já, eu tinha encostado nele mais cedo. Like this?
Yeah, any piece is fine.
Where you wanna place it?
In one of the buy boxes, maybe buy box 3. Yeah. So buy box 3B.
The part that falls, that collapses just.
Yeah, anyone is fine. Whichever is easiest to get to. Right. Collecting a piece of rock. Not yellow.
It will be too hard. It's like moving a lot.
Yeah.
Super tiny.
And it's gonna go in buy box 3B. Well, we'll see where it goes, but that's the intention.
Okay.
This one?
Yeah.
A rock sample is just
Biology.
oh,
Why not
biology.
put stuff?
Sorry?
No.
Yeah, because it might have
Life on it.
things. I mean, I had a rock sample.
Delete it.
I knew you needed rock.
Right. 3D.
It's the same. Hey, now our sample. Yeah, exactly. Perfect. That's why it's too fine.
Mm-hmm.
That's only same.
I think we're getting an amphipod as well.
No.
One.
Yeah, that's good. Yeah.
Thank you.
Thanks very much.
That was 385.
Thanks. Okay, so now we're gonna go back to exploring.
Right?
Nine?
14 and then 9.
Okay.
To that waypoint, please. Yeah.
Thank you. Is the light there a little too bright or something that is on the sediment, or is that normal? is it just a sediment or? Or maybe we need to zoom in a little bit, just a tad. Yeah, that's good. Oh, yeah, right. Okay, so we've done a bit of sampling and we're gonna continue with our exploration. We see that red thing on the seafloor. Well, that's attached to that stalk is a benthic ctenophore. Then we've got a shrimp, ooh, and a skate as well. The skate.
I think it's the same.
Yeah,
well,
probably.
they recorded it.
Yeah, that we saw earlier.
Here.
Yeah. Oh, the same one.
Yeah, but,
Oh.
but we didn't get a really good zoom.
Oh, so we can get a good zoom here then.
Well, it's not the one I saw.
No?
'Cause this one is a female. The one I saw was a male.
So this is the
We want
female.
Side view, front view.
Side is better, and the zoom in all the body, between the eyes, the tail.
So we're gonna do like a pan
Not just really, yeah.
or just get everything
zoom
in.
in,
Yeah.
Because it doesn't have the claspers. Oh yeah.
The claspers are the extra two things that are usually done there.
Yeah.
Okay.
It's like that. But
So this is a female. The males have things known as claspers, which are, I guess, two like extra, maybe less appendages.
it's a modification of the pelvic fins that the males have for reproduction.
Okay, so it's a modification of their pelvic fins that are used for reproduction. There it is. She's going.
It's okay.
It's good? Okay. Well, that's fine.
So this is sedimented. We can take the push course here.
Can't or can?
Can.
So, but before we do
But-
that,
Decisions.
what are your thoughts on what we have seen?
Speaking with Rob and Rich, like the main objectives should be these ones.
Yes,
Here.
that's what they just told me
So.
about.
we
So
can
extend
try to
for another hour and try to get to two, those 3 points around two.
Okay.
Yeah? Okay, let me put another shift on for everybody.
So we're still gonna continue there, or are we gonna
Yeah,
make our way
sounds
out
like,
there? That was 9?
8 and then.
8 or 9?
Oh, yeah, sorry. 9
9? Okay.
and then 5. Right? Yes. Yeah, our objectives are going to be these ones here. Like, let's go here, here, and we want to see these two. So,
Okay.
Yes.
And you can see all the marks and tracks in the sediment that indicate how active the seafloor is. So it's been quite an exciting dive today where we've seen quite a lot. We've seen skates, we've seen lots of mussels and tube worms and seepage. So what we're doing now is we're continuing to keep our eyes peeled for bubbles that are coming up from the seafloor. Because while we were conducting mapping of the seafloor, we would have seen quite a lot of indications of bubbling from the seafloor. So we're here to visually confirm those bubbles, and we've seen a bit, and we're hoping to see some more. I always get excited when we see like something dark in the distance, and then it's just sargassum seaweed. So yeah, like I guess people, you would assume that with the organic fall of sargassum that's coming down to the seafloor, that it would have caused like a frenzy, maybe a feeding frenzy, but in most instances we haven't really seen much interaction with the sargassum. seeing quite a few brittle stars scattered around. Nice tripod fish there. Yeah.
Wow.
So we've seen two species, two types of tripod fish so far on this expedition. One, this dark one here, and then earlier ones we would have seen like a light and dark colored patchy one. And they would have had like different fins as well. Fins that were more, how do I describe it?
Wow.
Less like whiskers and more like parts of an umbrella. And they tend to face into the current. Yeah, that is nice.
Wow.
Yeah, that's good. Thanks.
Maybe like shadows, but they rely more on the sensors that have all over. Yes.
That's a recording in there?
Yeah, I think we got some photos. There it goes. I think it might be another species from the one that we've seen earlier. The ones that we've seen earlier in the expedition.
Yeah, I think maybe we have seen three so far, right?
Right?
3 different tripods. Right?
Oh. Okay. I've only seen two. The black and white one-ish. Oh, I missed another one?
I think it did. I mean, not sure, but
That was a halosaur that just went by. And now we're seeing some benthic ctenophores and sea urchins. So
See, it's like that from
Cuéntame.
just making our way to these.
yeah, I think
Those
like
three
past
months?
Yeah.
And we're like hitting the points
dive
in that,
maybe,
right?
because we started
Um,
one.
so that's
I think they were
yeah,
fine,
yeah.
but I'm not sure if they followed
We'll
that
just
path
making
if
our
they
way.
went.
Alright.
Yeah.
The whole like collecting worms and things is over.
Okay, everyone, that's it for me. I now hand over to my colleague Giuliano.
Ah, los soportes. En los 3 puntos. Ok, thanks. Ahí estamos claros en los puntos. Ya. Salve. Tudo certo. You all thought you're gonna get rid of me that easy, but me as the cucumbers, I'm always back. So I'm back with you for the 3rd time today. Hello, hello. Olá todo mundo. Se tem alguém no Brasil ou alguém nos países de habla hispana, por aqui estamos. En portuñol, Spanglish, desde el Falkor 2. Alright. Terceira vez na cadeira hoje. Ah, planos mudam. Salud. Ah, porque... Não, tá de boa. Mesmo assim a gente tá à procura das bolhas, né? Então a mais alto dá uma melhor visual. We are in our last effort to search for bubbles in this area. We found, for those who just logged in today, we've actually found multiple seeps, maintained seeps during this dive in this different points exploration, to the point that we've actually extended our bottom time by, I believe, 4 hours by now. So exciting things to come in this hour, let's see. Para aquellos que nos sintonizan ahora, llevamos casi 4 horas de extensión del buceo, porque hemos encontrado bastantes seeps de metano en este buceo. ¿Cómo es seeps en español?
Emisarios.
Emisarios. Emisarios de metano. ¿Qué tal? Acabo de aprender una palabra nueva. Entonces estamos en esta última hora en un último esfuerzo para encontrar otros emisarios de metano. deveríamos estar chegando, né? Bom, é. A gente tá bem no ponto, né? Aí na frente tinha sargaço. É. Vamos ver um pouco para a direita, só para dar uma de boa. Clamshell on the right. Dime.
Los primeras que están las veces.
Y volvemos a esa.
Solo por aquello que no sé, cuando pase y quieran que vayamos a este.
Ya.
Creo.
Plastic bottle, yeah, looks like it.
Swimming to grab a opinion.
Yeah.
¿Cómo se dice foto en portugués?
Foto.
Foto.
Got it?
Anthropogenic. Yeah.
Well,
Oh. Ah, no, that's not the cash.
let me go down.
Yeah.
Got it? Thank you. Podemos continuar. Okay, so just a bit on the right, and then we'll continue to this one. So we'll try this one, and then we can go back, just to go to the furthest away.
You want to go to the further first?
Yeah. Yes, please. Unfortunately, in almost, if not in every single dive that we do, we find human trash, desechos humanos en casi todos los museos. Oh, let me see. That's what it was, exactly. Podemos Antes de continuar, dar a volta por aqui. To see what's in the other side of that. Actually, can you zoom in here quickly? Valeu. Can we get the lasers? Are they on? Oh yeah, they're on. Perfect. Ah. Oh, look at the size of that tube worm. Oh, it's aqui, ó. Oh no, that's sargasso. Hmm?
Let's zoom in the fish.
Let me zoom in the fish fellow. Beatriz is gonna tell us who it is. Díganos, Ana Beatriz. Filiforme. Filiforme. So cool.
Some sort of
Coscalish fish. Beautiful. Oh, and it's gone. Thanks, guys. Alright.
Sorry? Yes. There.
I keep confusing sargassum with mussels. Or maybe I'm like so expecting to see mussels. The other thing that we've seen in almost every single dive is sargassum. And sargassum is a natural occurring organism of the Caribbean and the Atlantic in general, this region. There's even up north Sargassum Sea, or the big sargassum patch. But on the last 10 to 15 years, we have seen a substantial increase the amount of sargassum, and that has actually been a problem for many of the Caribbean islands that rely on tourism because sargassum washes out on the beach. So if you have gone to the Caribbean over the last 10 years to vacation, or you live there, you will familiarize with beaches full of sargassum. And we've seen them a lot here in the deep during this expedition, to the point that we've actually done some experiments in past dives to understand, sorry, to understand better the carbon sequestration and content of sargassum in the deep sea. Para aquellos que nos han acompañado todos estos días y estas semanas, hemos visto bastante sargazo en el fondo, y a pesar de que es un organismo natural de la región, incluso en el Atlántico está le llaman el mar de sargazo, pero en los últimos 10-15 años ha aumentado sustancialmente la cantidad, lo que llega incluso a ser un problema. Aquellos que han ido de vacaciones a las playas caribeñas o que viven en ellas lo sienten, lo ven, el sargazo en la playa. Entonces aquí también lo hemos visto mucho en el fondo, en casi todos los buceos lo vemos. Some clamshells. We're following the path. Sea cucumber. Haven't seen one of those in a while. We're right on it. So can we do a bit of just a little short look around? Oh, skate. Want to zoom in on the skate, please? skate and a shrimp. Beautiful skate. Are the lasers just lost with the zoom? Oh, okay. Yeah, that's okay. Seguimos entonces para el próximo punto.
Sí, pero...
Estoy dándole una buscada
sí.
en la zona.
Even more zoom. Nice, and then this part, and then the tail.
This part here?
Yeah, all it's like you have to see the shape of the thorns to actually identify it.
The shape of the thorns?
Yeah.
Wow.
Yeah.
So
So as much as it.
Beatriz Naranjo is telling us that in order to identify species, one key component is actually the thorns in the tail. That's why we're doing as powerful as a zoom as possible. Mi colega Beatriz me acaba de informar que para identificar la especie un componente principal son ferns, espinas, las espinas de la cola. Entonces necesitamos calcular, por eso el detalle de la cámara, eso nos va a ayudar a identificar. Y con nos me refiero a ella, porque yo no tendría esa capacidad. You want to get closer to see?
You can try.
Oh.
They've been really.
Oh.
Say that? Yeah, they've been running away from us. In this side of this.
I don't know why. We're so like small and silent.
Sí, es como que Carlos hay que contar las líneas de espinas, sobre todo la línea del centro, la forma, pero es como que en los adultos la tercera fila, la fila del centro de la cola,
Ajá.
a veces empieza a perderse. O sea, como,
¿Se mezcla o qué?
como que cuando son juveniles la tienen muy marcada y luego la última, entonces
O sea, ni cómo ayudarles.
es como muy complicado. Ah, sí, much better. Great.
Yeah, he's zooming in now. Whoa.
Yeah,
There we go.
that's what we need.
It has a. Wait, is that like a.
It's like a sediment.
Oh, it looks like a.
Beautiful.
Este bro de ahí. Is that a Henry? Cangrejo ermitaño parece.
See the
Yeah, the tail part.
Wow, the detail is amazing.
I see 3 lines in there.
Yeah, yeah, this is different from the one that we had in the other
You wanna dive?
Methane seep, remember?
Mm.
We have a bunch of them. This
Yeah.
one is different.
That's been a leak.
for sure.
That's okay. Mm.
And yeah, the clasper splits the
Mm.
that's yeah.
This fits.
Nice.
So this one's a male.
This one is a male.
Yeah.
correct.
Male skate.
Okay. Yeah, I think we're good.
You wanna see the end of the tail, or you good?
No, I think with the other one we are.
Okay. Thank you guys.
Happy?
Happy like a hippo.
Average RV, weight updating it's to the northwest.
ROV bridge, repeat your message, Manny, please.
Waypoint's updated. It's still the northwest of us. So, stop.
Yeah, got it. Thank you.
Oh, now it's going. We can almost play a game, no sarcasm.
Yeah. And I've lost almost all.
Hmm?
Sorry? Ah, the cool kind. Okay. Something else. That was It's like there's a lot of sediment.
Okay.
So it's tricky. Yeah, they have to be.
And shrimp. Fish. Is here? Well, we can see if there is anything here. It looks like, but it looks sort of a bit. You focus in this part. Some worms.
These are
Hmm?
polychaetes.
Oh, look at worms. Some worms. Thank you. Thanks, Paul. Beatrice, do you wanna head there or there?
No, I think we'll stay around. I see that who's dead.
Yeah.
You are not seeing. No, looks like plain, right?
Yeah.
No, I think we will. We have to take the course. So we will look for a place where we can get those. So maybe just continue towards.
Fall.
Yeah, we will stop to look around here and then we will stop. We
To
will
get
try
the course?
Yeah, to get the course, but we will try to stop close to a benthic ctenophore to also collect the benthic ctenophore. We've been seeing a lot these days these stalks that have a red gelatinous animal, like that one over there.
Like this one?
Yeah, they're everywhere.
Okay.
We've been seeing,
So you want this one?
no, this one because it will be better to stop in a place where we can collect the push course and the ctenophore.
Okay.
Virtual here to continue
Can
with.
we walk just for a minute?
Yeah, copy.
Yeah.
I don't know before the wait for.
Can we see here, please?
What is that?
A long sea cucumber. We have seen it before earlier in the other
Yeah.
dive. There's so long.
The same ones that we have.
Yeah, the same one that we saw earlier.
The green ones.
There's two of them actually, I think.
Yeah.
But like soil here is it's almost like volcanic in color. Thank you. Put some we'll zoom on the right since we're already here.
Bridge on the video.
Just to look,
Out of the bridge.
look a bit on the right.
Sorry, can we pause it?
Sure, no problem.
Too soft sediment. Yeah. So good.
Sorry. Oh, that would be cool.
Bridge, I think you're clear to continue to move up.
Copy that.
Por supuesto, ahora que lo buscamos no aparece. I think this is one.
Lasicomorphy.
Yeah.
Aquí,
This
no?
one, yeah.
So we can stop here and we can get the
Yeah,
push
push
course
course and
and
we will
this
collect
guy right
these
here.
guys here. Yes. It's other, yeah.
Bridge, I think. Can we pause and move, please?
Holding now.
You want that little guy in the cork with it?
Yes, please. Yeah, the animal itself is like like they are making one more than one unit, so you need to grab the whole thing from the base. Yes.
Reach that thing with a port on.
Nos zoomos antes.
Sí. We want to do some zoom-ins too, so reposition ourselves. ¿Qué creemos que es esto?
Eso es un tenóforo bentónico.
Tenóforo bentónico.
Un peine de mar, como los que hemos visto, que le
Un
acercan
peine
y
de mar.
se ve como licencia.
Como los que... Ah, ya me habéis dicho.
Entonces, la
Tenóforos.
mayoría son pelágicos, pero
Right.
algunos han perdido su vida pelágica
Y se vuelven.
se establecen como especies de fondo.
Tenóforo.
Entonces, esto lo que hacen es que este tipo exactamente, lo que hace es buscar un coral o un tallo de
Una
esponja,
estructura, ya.
algo para estar más alto, y tiran unos tentáculos con los que atrapan
En la corriente.
comida. Exacto.
So this is a technophoro. And you see sometimes we're navigating and there's a jelly-like organism that is floating in the water and has soul bioluminescence. So it's the same type, different species, and they're normally pelagic. But this specific species has lost its pelagic component and has become benthic species. So it looks for coral or a coral branch or a sponge branch to be a bit higher in the water column. And then it extends its tentacles in the water to collect food as food drifts. Technophoro. Which we will collect now before the course.
Wow.
Yeah, can we get the zoom and the focus on it, please? Yeah.
Oh,
All right.
ves, son los
Beautiful.
tentacles
And that's
que
the
están-
tentacles that we were saying. The white ones. That's how he collects food. Esos, los hilos blancos son los tentáculos. Que es lo que con lo que atrapa la comida. Hmm? All good? All good on the pictures. Thank you.
I didn't get it.
Ustedes se ven bien, igual van a visitar.
Ah, ¿sí?
Yo creo que es la
Sí.
pantalla donde lo está viendo. ¿Sí?
¿Está hablando de este o del Ignore? Wow. Ya. ¿Es uno o son dos individuos?
Yo creo que hay más de uno, por lo menos tres tal vez.
So you wanna take the whole, like, todo el gancho, ¿no? Todo el palito.
Sí, no, ya sabe, voy a tomar de la base.
Yeah, so you're taking from the base, man? Cool. Yeah. Yeah, I think it's probably like multiple organisms. I agree, at least 3 probably. So we'll take it from the base. Wow, that is very deep root.
Interesting.
Wow.
It's so young.
And it doesn't break, it just keeps going.
Wow.
And it keeps going. Oh, I think it broke now. Somewhere? No?
Bring him up, Mark.
Oh, it's.
Oh. Wow.
Wow. Oh, shrimp. I love how the shrimps always randomly just.
Like the sponge, you saw the sponge? You took a piece of a sponge.
And the shrimp was coming.
And you were like 2 or 3 shrimps, like.
Yeah,
eating
to
the
eat it.
sponge.
Oh, there we go.
Thank you.
Perfect. Thank you, Money. there's a tornado in the back. All right. So that was river number 3. What's the number?
386.
386. Thank you.
Okay, now the push goes. No.
Oh. There we go. Cool. Now the push course, right?
All of them are all the biological ones are Yes, and then we will here, yeah.
There we go.
It's a sediment and it's Niskin and that's it. Because they took away the rocks.
Tengo que anotar algo para los push course.
Sí,
Ok.
el número, donde, o sea, el número de muestra y a dónde va cada uno. Pero al final, o sea, ahorita nada más los van a poner.
Ah, son
Y luego lo que
estos,
importa
¿sí?
es a dónde regresan y el número que traen y el número de muestra. Entonces, digamos, este es 1. Entonces, ahorita no importa, pero
Cuando los
luego,
regrese,
o sea,
¿dónde los
uno
va a
vuelve
poner?
aquí y es el 3
Ya.
para abajo.
Okay, the push goes here.
Yeah. Yeah, please.
You put it
That's
up.
good.
You need a picture?
You have a picture of that?
Frame rabbit. Don't be frame rabbit.
Yeah.
And then 12.
You could put it back.
It's all good.
That's 1.
It's all good. Next one.
Yes, just to enter. I think we will put them back.
Oh, you're gonna just put it back directly. Guess there's only 2 of them.
Nice. Looks so different.
Wow.
Looks so different from the ones from yesterday.
Yeah, that gray looks almost like volcanic to me.
Or mangrove. Really anoxic.
What number do you have?
387.
Oh, yeah. Thank you. And it was score 10, right?
I think it's
1?
1. Yeah. Estaban pegados, estos tenópodos. ese perfil. O sea, no son gusanos amélicos, sino que son gusanos.
Era un, claro, era un gusano, por eso
Es un gusano, sí.
se pronuncia el
Como gusano enorme. O sea, seguido.
Debe haber seguido
Sí.
kilos de metros. Y una parte viene con nosotros, así que algo veremos. You got the picture? No, nada más va a pasar.
So this was push one, push two. Let's see one.
Y ahora vamos para acá.
Sí.
What's next?
All right, that's it. We can finish the dive. Finish the dive.
Sí.
Thanks, Manny. All right, everyone. Thank you all for joining us. This is the end of our dive today. We had oh, look at that. We had a goodbye fish in the camera to say goodbye to everyone. And we'll see you all tomorrow. El fin del buceo de hoy. Vino el pez a despedirse. Estaremos de regreso mañana. Estaremos de volta amanhã. Boa noite, todo mundo. E obrigado.